as you can see. How many donors are available in each hospital or each city or each state or each country, right? And how many type 1 diabetic patients are in the requirement for these pancreatic tissues. So when you look at the ratio between the availability of donors and the list of the donors. And also, one more important challenge that comes into picture, if you go back to your basics of, let's say, immunology, for example, if you guys remember, we can't take anybody's organs and then inject into your bodies because everybody's immune system is different. So if you take a donor's organ and an organ, basically. So in order to avoid those complications, most of the time what these recipient type 1 diabetic patients, the immune suppressive drugs, basically, over the periods of 1 year or 2 years. So that is again another complication, because when you take these drugs over periods of 1 or 2 years, you're suppressing your own immune system. So that means you're already susceptible to having infections, cancer, anything like that. So that is again leaving opportunity for finding more treatment options, basically. So again, just giving you a brief, I will be little going faster from here. So again, giving you some background idea here. For example, if you look on the top panel, if you guys can see my cursor or not, just let me try to find laser point if I can. Yeah. Okay. So if you look into the top panel, so this was shown in case of artificial pancreas versus the islet transplant study. You might not have the clearances to do. So technically, you can form an embryonic, like a human embryo from those stem cells, right? So that's why those kind of issues are obviously possible, as you see in this diagram, right? Basically, as I said, we are getting the cells from here. So I want to use these cells for making some other cells like pancreas, but somebody can claim that I can make a complete human from those cells like this, standing human right here. So anyway, so stem cells. So technically stem cells are pluripotent. They are self-renewing cells. That means they have the capability to form their own cell type by highly proliferating. At the same time, they can form into any other body cell type like neurons, blood cells, bone cells, cardiac cells, epithelial cells, fat cells, any cells, any bodily cells, basically. So again, you also have adult stem cells in our body. For example, even in adults, specific organs like brain, bone marrow, these skin, for example, hair follicles. These are the specific tissue places where you can find adult stem cells as well, that have limited capabilities to form into specific subtypes. So what is interesting is, I don't know if you guys can recognize the face here. I have put it, most of the people. So he is Professor Shinya Yamanaka. He was awarded the Nobel Prize in 2006, and we are right now, after 20 years, 2026. Recently we celebrated 2 decades of identifying Yamanaka's factors. Basically what he magically did after a lot of research is he found 4 transcription factors which I have highlighted here as OCT4, CMYC, KRF4 and SOX2 that are sufficient enough when you add to any of these adult differentiated somatic cells that can reprogram the cells back to embryonic stem cells. That is something called induced pluripotent stem cells. So right now in the field of stem cell biology there are 2 terminologies. One is embryonic stem cells, which are always coming from embryonic tissues. On the other hand, you have induced any other somatic cells. For example, you can take a skin cell, you can take a epithelial cell, you can take a blood cell, anything. You can convert back to a induced pluripotent stem cell just by adding these 4 factors. famously called as Yamanaka factors. So, for which this guy was given the Nobel Prize 20 years ago. So basically, but again, I had the fortunate to attend one of his seminars in Bangalore when I was doing PhD. So his opening statement when he gave the seminar was this famous dialogue. The big question is, I found the answer to the dream of converting any cell to stem cell, but is that enough? Can we use these human stem cells to benefit for the society or to treat any diseases? That's what his statement was at the time, which is what bringing to us again 20 years later, which are also we are trying to address here today. So again, just this is too much of information in this slide, but just to show you the reprogramming of stem cell fate is being right now done across the world in different labs, just to make stem cells to include dopaminergic neurons in the brain to treat diseases like Parkinson's disease, or for example in our lab we are doing the iPSCs or embryonic stem cells to differentiate into pancreatic cells, specifically beta cells that can secrete the insulin so that we can treat the diabetic patients. You have other cell types also. For example, you can use this for cancer therapies. You can use stem cells for eye diseases, age-related macular degeneration, cornea damaged cornea, or muscular dystrophies, anything, any sorts of diseases, basically. So the whole field of regenerative medicine is exploding right now with the developments of stem cells. So yeah, so basically when it comes to stem cell, what is mainly important is they basically depend on very large complex pathways of signaling molecules. Okay, those are called extracellular matrix, and the signaling pathways of surface proteins and intracellular transcription pathways. So the interplay between these two is very critical when you want to make these cells to differentiate into any other cell type. That's why I just want to make sure you know. You don't have to know all the details, but it is a very complicated pathway. But when you want to make a specific pathway, you have to pick and select some of these specific molecules, and you can work on that. So going back to the pancreatic tissue, again, we are interested in pancreatic tissue here, and I am highlighting here again. We are interested to make alpha cells and beta cells that secrete both glucagon and insulin. Those are the hormones that are being secreted from pancreas. And main research focus in our lab at Michael is basically to make the stem cell differentiation towards pancreatic lineages. And once we know the process is possible, we want to make this process robust and also scale up. For example, how quantitative we can do this. How many liters of media, how many millions, how many billions of cells we can make in a single day, or in a month, or in 2 months. Like for example, like you guys heard, like in COVID, we all had challenge of COVID vaccine. We all had the demand of COVID-19 vaccine, for example. When it comes to that level of demand, can we make these kind of cells that demanding, for example? So we are finding solutions for those by using bioreactors, different kinds of bioreactors in the lab. And also we are using analytical platforms like flow cytometry, PCRs to check for the qualitative readouts whether we are reaching our goals here. And also we have recently started to implement machine learning and artificial intelligence tools to find the future integration algorithms to know better outputs for the differentiation of stem cells into pancreatic. The dark shaded things that are called, those are the pancreatic cells we have. We are using sheets, we are also finding pouches. We are making intravascular devices also. We are using all these type of biomaterials in which we are adding cells again. For example, all these yellow color things is the 3D printed biomaterial, and the cells are those colored ball of cells you see in your pink color. So when you have this 3D printed material with cells, you can imagine that it is something like your organ in your body, where your blood is flowing and then your cells are surrounding it, right? So it can get your oxygen, it can get your nutrients and everything, and it can survive and it can protect from immune system as well. So this is the bioprinter picture we have in the lab, and this is something the bioartificial pancreas we are trying to do in the lab, for example. So you can see in the bottom, we have those channels printed here. This is how it looks, and you coat it, and you can, and then we perfuse this for almost like 2 weeks, 15 days. Media from the bottle goes through these pipes, goes through this chamber, and the cells are sitting here, and spent media comes out, and then comes out. So this whole cycle happens 24/7, no stopping, for almost 15 days. And then on the day of 15, we take the cells out and we check the insulin response. They are showing very nice insulin response. So this is how we kind of try to develop our models for our next goods. So what I'm trying to tell you here today in my talk is
good day, everyone. I am Shrestha Ananya, and I am currently studying my third year MBBS in Trinity State Medical Academy, Bishkek, Kyrgyzstan. My paper that I have presented here is on resistance and AMR, and also how integrating probiotics along with innovative drug discovery can help combat a very growing crisis in the field of medical sciences. And I would like to first say why AMR is such a big problem. Antibiotics have transformed modern medicine for us by making almost all of the deadly diseases treatable. However, at times, overuse and misuse in the fields of humans, animals, and agriculture has made the very microorganisms that we have been studying on for years resistant. So, in infections with multi-drug resistance, bacteria are very hard to treat. So, it generally leads to we have some. Having said that, I would like to offer my sincere gratitude for everyone, for your patience, and also Dr. T. Rosemary and her team for giving me this wonderful opportunity. Thank you so much.
University. He published Seed and Quality of Grapes. Dr. Co-Nobel Laureate, France. He has significantly contributed and is committed to the furtherance of agriculture. Now,
non-destructive non-destructive techniques. So because we were seeing nowadays is mostly this. So is there entire lot is being rejected. Like recently the European has rejected, even the US has rejected because of these issues. So this quality check is also difficult because you cannot cut open all the fruits. No, when sending the container, if it is 1 lakh fruit with the frost, even when you store the fruit at lower temperature, sometimes the fruit gets frost damage. For example, mango you cannot store test every fruit. I mean, check using penetrometer test. And for TSS, like the sugars, how much it is present, all these parameters will give the freshness and quality. So these using TSS using the refractometer and some of the other chemicals by destructive, all these are destructive techniques, right? So that cannot be done for all the fruits. So which can be using any other solutions means these are the solutions that can be used for non-destructive like NRI spectroscopy and MRI, NMR technologies, and EROS and ETANC. These are the latest techniques has been adopted basically by the industrial and the ultrasonics and hyperspectral imaging system. So whatever the commodity is being produced, either in the domestic or international market, is being assessed by these techniques. Then only we get the certification. So that certification can be used by the external agencies like exporters or importers of other countries. Then only your commodity is being exported. So you should take the license of these things and certification, then you will get it. So first of all, these are the commodities, the methodology adopted, the different methods. One is optics or X-ray method or mechanics like electromagnetic system. These are the different machineries, different techniques has been used to assess the quality of an individual fruit for different components. For example, optics system if you use, so you can assess for the shape, size, color and external defects also. Like a machinery, whatever you are sending the sample, the size also grading can be done according to the size. For example, I need the best grade one, like for example half kg or 500 grams. Scan like the human body we do X-ray, like CT scan system. Similar, all the commodities has been done and industrially they are doing. Otherwise, our commodity gets rejected. So that has to be the one of the standard operating procedure SOPs of different countries. So this system, when you pass the commodities using that the system inside where X-ray is there, you can detect the internal cavity whether it is damaged or not, whether it is ripened or not. The proper ripening should be done. So all these procedures can be checked using these systems. So we have some other techniques using mechanics, sonic sound system, ultrasound system, and right? Same that can be checked in a bulk quantity using impedance or MRI technique. So moisture level, because it is passed in a huge quantity that cannot be traced. So individual rice you cannot trace no individual grain. So entire lot standards, all the quality checks can be done using a single time technology. So irradiation technology in that nanometer, and that can be great advantage to find the many parameters in one single stretch. Quality and rich in antioxidant and it is anti-aging property for your health. So all these things can be detected using some of the fluorescence technique of this machinery. So that can be used. So you can measure within 1 second. You can measure the length of a light, and the same acoustic or sound where ultrasound can be used and to see whether it has matured or not, whether it has any defect that can be irradiated to the fruit, and even the pest which is present in the any fruit or your produce what you are making like sauce and juices or anything, even if it have any bacterial content that can be killed using this irradiation technique. That is not harmful to the human level when you use a certain level up to the quantity level. Like we have a cobalt 60 and it is commercially being used, and it is not harmful to the human being if you consume it up to certain level. When it gets exceeded, that was harmful to human beings. Okay, like this imaging of individual fruit, you can see how they have taking as X-ray imaging. So like in the airport when you are going to all the baggage, everything is being detected now. So what is present inside the fruit. And even if you carry a plant, okay, from the airport, if you have a soil in the bag, right? The soil may contain a lot of bacteria and everything. So when you transport from international, they reject your product. If you transport domestic, they may accept because within the country. If it goes out of country, they won't accept it. Because of that only plant quarantine is existed now. Indian plant quarantine, they check all these things because you may introduce some new pest to some of the countries. So that is why that detection can be done all these like X-ray imaging systems. So same, all the samples of the fruits in industrial industries, they check like this system, like CT scan. So even the fruits, within the fruit, even the seeds being accepted by any country, except some Middle East countries, they accept it because most of the Indians consume these onions. So we accept. But if you go to the Japan or So still it is there 20 more slides. So we can continue the later in the after post lunch. So that I'll take you in another session also. Meanwhile, sir has told me that one more speaker is ready, so we can continue with that. Okay, right? So it is almost about to complete. The picture is the new techniques. What is there? Just I want to introduce you.
Anyways, good to see all of you. And this lecture is particularly made in conjunction awareness on some new topic, which is not there in the books. I work on this topic basically. So this is something related to microbial signatures of diet, understanding the host microbial interplay or microbiome interplay. So what happens between the microbiomes and the diet, whatever we're eating? So how does it metabolize? What's the biochemistry? What's the metabolism behind all this thing? So I humbly request everybody to keep it silent because I get a little irritated if you make noise because we have come from long distances to deliver a lecture. So I hope other people who come here also come with a lot of immense scientific and intellectual capability. The human genes, nearly 26,000 to 30,000 genes are there. So if you talk about plant genes, there are nearly 46,000 genes. So what happens is like genes compared to the human beings, the plant has got more genes. But when you look into here, the human gut microbiome and human tantamount to more than 100,000 genes, maybe more than the size of the plant genome, plant genes. So in contrast with the first spool of the immune cell. So they are essential for human health and influences the gut-brain axis. They support the long-term homeostasis. The major area of our modern medical research. This is where the translational research is going on, and most of the future medications will be mostly based on microbiome, metabolism, metabolites produced by that. So look at here, a human gut microbiome, our internal unit.
Good afternoon, everyone. This is Mukesh. I'm coming from Raghu Engineering College, Visakhapatnam. Today I will present. I am presenting my research on AI-based early cancer detection using blood biomarker data. These works focus on machine learning techniques to predict potential cancer risk using cancer diagnosis. Transforming healthcare with AI, comprehensive blood marker analysis, machine learning, algorithm comparison, performance evaluation metrics, results of machine learning performance, impact on preventive healthcare, overall conclusion and question answers. Then come to disclaimer: these views and opinions are not are those presentation of the others does not necessarily reflect official policy in the or any organization. In introduction, cancer is one of the leading causes of death worldwide. Focus on preventing early errors. Overall conclusion: AI enables accessible non-invasive cancer screening using routine blood biomarkers, and also it is a powerful routine screening tool by combining multiple.
Good afternoon, everyone. This is Rupa from Raghu Engineering College, Visakhapatnam. I'm presenting my project, Personalized Nutrition Recommendations using Machine Learning. This is focused on how AI and ML can transform traditional diet into smart and personalization healthcare solution. My project agenda is first, why personalization nutrition matters and types of data I'm used, how to convert raw data to action. I generated my project analysis individual health data including age, body mass index, medical history, lifestyle and food preferences. I'm using machine learning algorithms like decision trees, random forest, KNN and clustering to generate personalized calories intake plans and balanced macronutrient recommendations. And continuously I'm improving by featuring in engineering and modeling. I'm using a lot of machine learning techniques like decision trees, clustering, deep learning. I'm also explaining recommendation outputs and personalization. The system provides tailored daily calories, macro distribution, and specific meal options, agreed safe distributions, timing aligned with activity, data guidelines, and outcome metrics. Potential impacts and early evidences are sustainable HCl 25%, average HbA1c, HbA, 1c reduction to 0.6%. User satisfaction is 4.5 out of 5. Overall conclusion is machine learning enables personalized nutrition, bridges data science and healthcare. Further steps, larger clinical trials and data EHR integration and scalable clinical in the loop workflows. Thank you.
Sir attended different types of workshops. Now he is the assistant professor in biotechnology. I am inviting Nadeem Siddiq sir to present or to deliver a talk. Show of your hands, all of you. Yes. Now I believe that you had a great lunch. Now let us dive into the main topic. That is thank you. Okay, engineering a tissue. Now, the question comes, why do you need to engineer a tissue? The only question which comes to our mind is, why do we need to engineer a tissue? Because there is some problem with the tissue. Now, why that problem will, in daily life we will be seeing more number of accidents which will damage your tissues. And the second one is cell, and third one is biological factor. For your information, in order to show you that I got the first component, because I cannot carry the cell and biological factor. This is nothing but a scaffold. This is nothing but a scaffold made up of natural polymer. Okay, let us talk about much more about these things. So the first scaffold. It is made. Scaffold is nothing but it is a temporary supporting structure for the cells to grow, proliferate, and differentiate. What is the function of a scaffold? It is providing a substrate, that is a matrix or a support matrix for the cells. Cells is nothing but the second component. They are giving a support to the cells to grow, multiply, proliferate, and finally differentiate. Now the second component is cells. In tissue engineering, we will use stem cells as second component. Why we use only stem cells? Because stem cells are the only type of cells which have the ability of both proliferation as well as differentiation. You can use other cells also, but they won't differentiate. Here, our main purpose is to differentiate your cell into any type of lineage. So suppose bone defect is there, that defect should be cleared by using bone cells, that is osteocytes. If there is a defect in cartilage, whatever the seeded cells, stem cells are there, they should get different. These biological factors. Biological factors are nothing but growth factors. Growth factors which assist in the differentiation of stem cells from one lineage to another lineage. See, we know that stem cells can differentiate from one lineage to another lineage. The capacity of this differentiation is known as potency of a stem cell. So based on its differentiation, they are divided into totipotent stem cell, multipotent stem cell, oligopotent stem cell, and finally differentiated cell, that is unipotent. It cannot differentiate further. It cannot differentiate further unless and until you genetically reprogramming it. Okay. Now let us go into the statistics of. So the main thing is, like by using this tissue engineering strategies, what you can do, you can minimize the number of deaths. And the donor crisis, whatever I said, donor crisis is there, that can be overcome if you can able to design a prototype in your lab and give it to the patient after a series of tests only. It's not like that today you are preparing, tomorrow you are today you are preparing and tomorrow you are implanting. It's not like that. Thus there are series of steps are there that I will discuss in detail. As I said, the three components include first one is scaffold, second one is cells, and third one is bioactive factors or growth factors, what we can say. See, these are all the general topics which I have not, till now I have not entered into the topic. Okay? Just I am giving you an overview of what I am going to speak in next 15 minutes. Okay? So there are different strategies of repairing a damaged tissue. It may be bottom-up approach or top-down approach. Throughout my PhD and postdoc work, I have gone through all these two stages in order to repair the damaged tissue. applications of this tissue engineering. As I said, tissue engineering is applicable from top to bottom. If bone is damaged, bone tissue engineering. Cartilage is damaged, cartilage tissue engineering. Myocyte, that is muscle tissue engineering. There is a damage in nerves, nerve tissue engineering. Likewise, each and every organ can be replaced, restored its function. Okay, that is the main motto of tissue engineering concepts. Now, what I said, the second component is stem cells, right? Based on the potency, that means they cannot give you, if you can able to isolate the cells from totipotent, it can give in the lab into placenta also, that you can differentiate, that you can generate a placenta also. Whereas for mesenchymal stem cells, if you are isolating a stem cell, so those mesenchymal stem cells are limited. Their differentiation ability is limited. It is like, when you are in school, you have multiple options. You can become IAS, IPS, doctor, collector, anything you can become. Once you have selected BPC, then your chances are restricted, because you cannot take now EC, CSC, computer science. Still after your BTech, after your graduation, then narrow down. So it is also like that. The differentiation ability of the stem cells is also time dependent. That means as you go from embryonic stem cell to adult, the differentiation ability of stem cells will be limited. It will be restricted. Okay? That is the second part. So in this second part, in this slide, what I am trying to tell you is the potency of a stem cell will be continuously keep on decreasing. So in order to use these stem cells, we can able to use, if you can able to use from that embryonic stem cell, it is highly useful, but it is unethical. We cannot use that early stage embryonic stem cells. Even after taking patient consent of the stem cells, okay, which are pluripotent in nature. Now, let us see that first aspect again. What is that first aspect? First aspect is scaffold. So this scaffold is, there are different types of different forms of scaffold. What I have shown you, this scaffold, it is a porous scaffold. It is having pores in it. If you deeply observe this microscopic structure, what I have shown you, this is that structure. If you see through either scanning electron microscope or field emission microscope or transmission electron microscope, you will observe a honeycomb-like structure. So this is a porous type of scaffold. Top two, these two are fibrous form of scaffolds. They are either aligned scaffolds or unaligned scaffolds. The third one, last one, that is hydrogels. All these are the different forms of scaffolds. So for these biomaterials, what we use are either natural polymers or synthetic polymers or ceramics. So natural polymers, they are abundant in nature. Their biocompatibility is very high. Their degradation rate is slow. Their degradation rate is fast, and they are widely used because of their biocompatibility aspect. When you come to the synthetic polymers, in the case of synthetic polymers, their biocompatibility is low, but their mechanical abilities are low. That means mechanical strength of a synthetic polymer is very high. So what are the materials we can use for repairing scaffold? Either natural polymers or synthetic polymers or bioceramics. In bioceramics, tricalcium phosphate, hydroxyapatite, wollastonite, all these type of things can be used. Now the scaffold material is ready, but we need to use certain method to fabricate your scaffold. So these are the scaffolds what we have prepared by using freeze drying. We used by freeze drying. It is also known as lyophilization, where your solvent will sublime instead of evaporation. Your solvent will sublime when you dissolve your polymer in its suitable solvent. After freeze drying, the complete solvent will sublime, leaving behind the porous structure. So these are the various methods. So nowadays, at most advanced methods are rapid prototyping. They are 3-dimensional, 4-dimensional, even thermoresponsive. See, due to the external stimulus, the scaffold is changing its shape inside your body. Imagine you have kept a scaffold inside your body. Whenever there is some increase in acidic juices inside your tummy, then that scaffold will get activated and starts releasing certain drugs, which will decrease the acidity. That is good, right? So that is known as 4th dimension with respect to thermal stimulus, with respect to pH stimulus, with respect to time dependent. So they will fall under rapid prototyping scaffolds. But what I have used, they are not of rapid prototyping. They are of freeze drying methods. Okay, freeze drying as well as freeze. That is what the scaffold is. What is it bringing, right? So this is the morphological characteristics of scaffolds, what we have prepared by using a natural polymer known as chitosan. Chitosan is derived from chitin. Chitin is extracted from the crustacean shells. Crustacean shells, the mollusks have this chitin. From this chitin, upon deacetylation, you will get chitosan. That is the main principal compound of my research area. On chitosan, only the properties of natural polymers, though they are most biocompatible in nature, their mechanical strength is low, relatively. Their degradation rate is fast. So what I have to do, I have to improve its mechanical strength. How I can improve? By using a bioceramic, by incorporating a bioceramic. What is that bioceramic? What we have used, that is tricalcium phosphate. See, my main target is towards the regeneration of bone. Bone is having the main composition collagen and hydroxyapatite. Collagen is a natural polymer, hydroxyapatite is mineral. So these two are the main components of a bone. So I need to mimic these two components outside the body environment. That is in vitro conditions. I have to create one template. I have to create a prototype. I have to create a scaffold which is supporting the growth, proliferation and differentiation of cells. That is my module. Okay, these are the microscopic structures, the size as well as dimension of tricalcium phosphate. What is tricalcium phosphate? It is a ceramic. So we have varied the dimension, that means in micro-sized tricalcium phosphate we have used, and second one is nano-sized tricalcium phosphate we have used. Out of these two, we have found that by increasing the concentration of this tricalcium, there are certain changes in the properties of the scaffold. In order to use it for bone, till 40% TCP you can incorporate into, chitosan matters. Beyond which what happened? Even if I improve 50 to 60 also, mechanical strength didn't get improved. See, you can see one compressive strength. See, compressive strength of this particular scaffold is 2.67 MPa. MPa stands for megapascal. MPa stands for megapascal. There are two types of bones, cancellous bone and cortical bone. Cancellous bone, for it requires 2 to 10 MPa, whereas for cortical bone it requires greater than 40 MPa. My particular target is to go for cancellous bone, which requires 2 to 10 MPa. So my mechanical strength is falling under 2 to 10 MPa. So I can go, can I use this scaffold for cancellous bone? Arginine, G stands for glycine, D stands for which amino acid? Any one of you? Aspartic acid. T stands for aspartic acid. So fibrin is rich in RGD motifs. What is the function of this RGD motif? RGD motif will attract more number of cells towards its surface. So what I have done, I have always, irrespective of the number of days, either it's a predetermined time point, 3rd day, 5th day, 7th day, 14th day, what's happening? On that scaffold the number of cells are more, because I told you, a scaffold with fibrin will attract more number of cells. Okay? That is, the content is also increasing, which indirectly indicate you that there is an increase in the cell viability. This is till polyphase. See, we have seen attachments, but after 21 days they have differentiated into osteocytes. But after 21 days they have differentiated into osteocytes. How I can prove that? By various assays. Like first assay is alizarin red assay. So in alizarin red assay there are alizarin red assays there, ALP is there, GAG content is there, total calcium deposition. There are huge number of assays, out of which main things I have kept in my slide. Okay, the first one is this alizarin red assay, which indicates that when a cell is differentiating, MSC is differentiating into osteo will express these hormones at varied levels. For example, you see this beta-actin, bone cyanoprotein, osteonectin, alkaline phosphatase assay, osteocalcin and collagen 1. These are all the markers expressed by an osteocyte. These are all the markers expressed by osteocyte. That I have confirmed through RT-PCR studies that all these markers are expressed. But on fibrin-coated scaffolds, the scaffolds which have fibrin, their intensity is more when compared to a scaffold without fibrin. It indicates that fibrin is not only promoting the growth, not only promoting the proliferation, but also differentiation of MSCs to osteocyte. Now, as I said, these scaffolds are the scaffolds which are made up of chitosan. They are degrade fastly. So you need to control its degradation. In order to control its degradation, we can go for certain cross-linking agents which are plant origin. One is genipin, which is extracted from geranium plant, and the other one is chemical soluble TPP, tripolyphosphate, water soluble reagent. That is also a chemical reagent. So, okay. Now, if you see that optimization of various concentrations of cross-linking agents. As I said, this is the morphological features. Now, this is very important. These are the confocal images. Confocal images of the cells. See, can you able to see this background? This background is nothing but your scaffold. And this, green colors, things what you can see, dots, you can see, this is nothing but cells, which are happily growing. See, the cells are, connected with each other. This is the Z-stack image of confocal microscope, which indicates the infiltration of the cells into the scaffold matrix. Okay? So up to what depth your cells are infiltrating. These are the, as I said, my continuous work is on chitosan and other types of polymers. This is the second unit, second topic, what I would like to conclude for 5 minutes. See, this is beta tricalcium phosphate, chitosan and PCl. Here I have used polycaprolactone. Polycaprolactone is a synthetic polymer. So it will further enhance the mechanical property of the scaffold. My aim is to prepare for 2 to 10 MPa, right? I got 2.67 only in part 1. But I can reach up to 8.9 also, that is also possible. But by using this PCM, the product into the market, you need to check whether that product is in vivo biocompatible or not. For that we have used ICR mice strains in both sexes, both male and female. We have observed their routine activities. After that we have given averting, which is sedative. Then we have inserted our implant. After 15 days, after 3 months, we have sacrificed that animal. After that, what we have seen, these are the images, histological images, where it shows the cells are, getting infiltrated into the scaffold matrix. So it indicates that your scaffolds are not only compatible in vitro, but also incompatible in vivo.
To introduce our next resource person, Dr. Ashok Chakravarti.
This is the last talk. After this talk, 2 more presentations from ALU. Then we will proceed to our valedictory function. Okay, thank you.
Good afternoon, dear students. It's a great honor and privilege to introduce our esteemed speaker, Dr. G. Ashok Chakravarti Garu. He is an accomplished agronomist and currently serving as an assistant professor at Malaradi University. He has previously worked at Loyola Academies Hyderabad. He holds PhD in agronomy from Annamalai University with over 7 years of combined experience in teaching and research. His areas of specialization includes resource conservation technologies, weed management, soil moisture conservation techniques, and sustainable maize and rice cropping journals, authored books and book chapters, and has actively contributed to national and international conferences. He remarkably has recognized with several prestigious awards, include Young Agronomist Award, ILSC, Young Researcher Award, and Dr. APJ Abdul Kalam Young Scientist Award. Beyond research and academics, he is deeply involved in organizing workshops, agribusiness summits, and international conferences, contributing significantly to agricultural innovation and knowledge dissemination. Today, we are privileged to have him with us. Let us warmly welcome Dr. Ashok Chakravarti Garu.
Good afternoon, respected dignitaries, students. Good afternoon, respected dignitaries, students, participants, and everyone present over here. I am Dr. Ashok Chakravarti, working as an associate professor at Malaria University, Hyderabad, and I am proud to say that I am the excellent of staff, not the student, where I worked at Hyderabad during 2017 to 19, and later. And currently I am working at the Malaria University. To present today is not about from my research work or any one of the research work. Just I want to give a talk on sustainable agriculture. The thing is here, nowadays in every family there is a mini farmer. How many of you agree? Each one of you are having a plant at your house, isn't it? A plant, one, in a pot. Okay, if you are having a garden, you are the small farmer, I can say. Instead of mini farmer, you are the small farmer. If you are having a plant at your house, where day-to-day life you will be watering it, or sometimes you will be giving the kitchen waste to it, and you will be trying to keep it under the sunshine, isn't it? How many of you agree? Even if you don't do, minimum your parents might be doing that, or your mother especially. So today I am going to present on the topic Sustainable Agriculture for the 21st Century, Challenges, Innovations and Future Pathways. So before I enter diving into the main core topic, I would like to give a little data about the agriculture so that the students, those who are from non-agriculture background, they will get to know how in individual life of agriculture is climate change, biodiversity and livelihood. If you look at each point, many of you know the food security. That means we have to meet the need of the hour. That is right. Currently we are around 140 crores, our Indian population. Right now we are having 364 million tons of food production. But as years pass by, the population is increasing, but our Indian land area is not increasing, isn't it? But population is increasing, and we have to meet the need for sure. We are forced to do that. How many of you know about the Green Revolution? Can I see the hands? How many of you heard the term Green Revolution? Who is the organic farming? So you know about the organic farming, and each and every farmer, they were doing organic farming, but whatever the food that we are producing in our country is not sufficient to meet the need. So that is the time where our Indian government has to take the bold step to double the farmer's income, at the same time to bring back the uncultivated land to the cultivable land and increase the irrigation facilities, that is the water. That's the time we have started importing the fertilizers and also initially cereals, that is rice and wheat. As you all know, South India mostly we take cereals and rice, and North India wheat. So they have to import all the cereals to meet the demand is going to rise by 60 to 70%, and at the same time agriculture uses 70% of the global fresh water. If you compare the ocean water to the fresh water, we are having only around 2% of the fresh water out of it, and occupies 38% of the earth's land surface and contributes 20 to 30% of the global green gas emission. So you might be thinking, what are the major pollutants in and around? We will be first getting to our mind that factories or industries, right? But do you know that agriculture crops also are causing the pollution? All of you know about the photosynthesis. You might have studied in your school days, isn't it? What is the meaning of photosynthesis? Taking carbon dioxide and releasing oxygen. How about the respiration? We do that. Taking oxygen and releasing carbon dioxide. And if you see that, plant also does respiration as well as photosynthesis, where photosynthesis will be taking place in the presence of sunlight, but respiration will be taking place throughout its life cycle, during the day and during the night. They too release the carbon dioxide. So now, if you see here, the growing population is increasing, at the same time the challenges are increasing, and we are forcing to convert back to the sustainable agriculture, where we will be much focusing on the organic agriculture. But at the same time, if you go for organic agriculture, what are the limitations that we can that are faced by the farmers that I will explain in the upcoming slides. So let us see the challenges. Why sustainability matters. Hope you know the meaning of sustainability in general term. So sustainability is nothing but we have to look after us as well as for the future generations or for the future. Both the ways. Not only as we never think about the selfish way, but we have to look back to our future generations in the upcoming days. So here, because of the chemical farming, already climate is greatly impacted and soil degradation is happening and water scarcity already started and biodiversity loss. For botany and geology students can give better answer about the biodiversity loss than me. The reason is how pollinators are decreasing day by day. Hope you all know, especially botany students, that for the pollination, who requires more? Honeybees, right? So, can you find enough number? When I see at my department, we ask students to go for the insect collection. Actually, I don't know about the journal and botany department whether botany probably herbarium plant species you have to place on the paper, and geology students may be collecting some of the species, important species. In our department, students will be roaming around the field. Standing water doesn't mean that rice crop requires the standing water. Standing water doesn't mean that rice crop requires the standing water. The reason is to avoid the weeds in the rice crop, they keep the standing water. The major reason, without standing water also rice crop can come well. Because of this water present in the standing water also rice crop can come well. Because of this water present in the rice crop is emitting the methane gas. You know that it is emitting the methane gas, and apart from that, the major issue, the running issue at Delhi is the pollution. How many of you read this news? The major issue at our capital is the pollution. You know the major reason? The surrounded by, we have two states, Haryana and Punjab, and farmers are just burning the crop residues. That means once the rice crop is harvested, the leftover debris which are in the field are burnt. Even the sugarcane crop, the leftover is burnt, so that immediately they can clear it off the field and they can go for the next crop. But they are not thinking about the consequences of it. So whatever the smoke which is generated from the farmer's field, the Delhi is affected so much. So that is a major carbon monoxide which is released to the atmosphere. So by which the greenhouse gases which are emitted from the fields might be methane gas or carbon monoxide or CFC, which are causing the ozone layer to be depleted, and finally the rising of temperature is happening. Even we can sense that as years are passing by, especially in the summer season, we feel the suffocation and the rising of temperature, right? And extreme weather events. So earlier, if you ask your parents during the rainy season that we call Kharif monsoon, there we don't find the rains at a time coming, and around some like 15 days or 20 days gap again another rain has come. But often we receive in splits. But at present, like few years, if you count, there we are finding that at a time we are getting the rains like cloud bursting. They are coming up with the term cloud bursting. At a time within the night, whatever the rainfall that's supposed to be received throughout the 2 to 3 months, we are receiving within a day. And again 2 to 3 months we are not finding any rain. So nowhere it is useful for the farmer. Farmer requires the rainfall throughout the season in splits. But here we are receiving the rain within a day or within 2 to 3 days. There we are not getting enough rainfall. These are called extreme weather events and shifts in crop productivity. That means because of these rains, farmers are not getting the enough yield. Maybe this year is getting the good yield, but next year because of the weather parameters is not getting the enough yield. These are the major challenges that are caused by the climate. If you see look at the soil health challenges. What is the meaning of soil erosion? Agriculture students. Loss of topmost layer of the soil because of the water. Okay, when rainfall occurs, when rainwater is moving along with the rainwater, soil also will be washed away, isn't it? So, if you see in general in agriculture, land, top 5 most centimeters will be having the major nutrients that are needed to the crop. So that's where every
along with the water, the soil is eroded from the farmer's field, along with the nutrient depletion is happening and loss of organic matter too. So this is another major drawback which is in soil health challenge. And water scarcity, every farmer nowadays, every farmer is having the borewell. And just farmer, once the season begins, once the time of sowing begins, throughout the season, he will be extracting the groundwater from the field, but farmer will never try to recharge back the groundwater. That's where we are over extraction of groundwater. And also inefficient irrigation. So we are having different types of irrigation methods when we look to the agriculture, like surface irrigation, subsurface irrigation and micro irrigation. Most of the farmers are going for the surface irrigation, where he doesn't know how much quantity of water to be given to the crop, just he will straight away give to the crop, and whatever the amount required by the crop, that amount only will be taken by the crop. The rest of will be lost by evaporation or leaching or whatever other causes. And by which the water is depleted and competing for water is increasing. Every year maybe hectares or 50 hectares or 200 hectares, and they are affordable to buy machinery. But in our country, most of our farmers are small and marginal farmers, where our farmers cannot afford the machinery, which are in cost lakhs. So that is one of the drawback. And access to technology. Our farmers are not well educated and they are unable to access the technology. But the good thing is nowadays some of the softwares they are turning back to farming. So there it is helping out. And income instability. 1 year they may get good income, but the next year they may not get again. So that is also one of the major constraints. And now let us look at the principles of sustainable agriculture. See now, till now whatever explained, these are the major challenges which are caused for providing the good food. Till now our government is focused much on the food security, but now time has come to focus on nutrient security. Hope you understand the difference between these two terms. Food security is different than the nutrient security. FSC students, you can work on it well. Earlier, we are striving to provide enough food to the growing population, and still it is consistent, but somehow we are beating it. But we are not helping the population to provide the enough nutrients which are required in day-to-day life. And economic viability, whatever we are providing to the market from the farmer, whatever is selling the produce to the market, it should get enough money, otherwise there is no use to the farmer. And social equity, for every farmer, government has to provide the equal opportunities. Same crop if you are growing, if you don't get enough water, we cannot blame the government, but instead we can straight away change the crop so that within the limited sources we can raise the another crop. And the another innovation is precision agriculture, where uniform field practices to site-specific decisions. For which, for your better understanding, I can give an example that every farmer, when he applies the fertilizer, he will be using the method called broadcasting, right? Straight away, he will be applying, just scattering the granules to the field. But farmer never knows in his acre land where the nutrient availability is more in that particular site and where the nutrient is less in the another site. So by using this precision agriculture, site specific straight away we can apply where directly they will scan the soil and they'll find out in this location nutrient is high. For example, nitrogen is high. Let us avoid applying in this location. And wherever it is low, there straight away it will apply the nitrogen. This is how we can reduce the application of nutrients. And one more thing, farmers, though we are suggesting to apply, for example, rice crop, apply only 150 kg of nitrogen. Never ever he will listen to us. He will apply 200 or 250 kg of nitrogen. It's just dumping the nutrients to the soil that he will get the bumper yield. No, whatever the crop requires accordingly it will take. Beyond that, crop will never uptake. That will be settled in the soil and we are destroying the soil health. And sensors and GPS, reduced input use. These are whatever I have explained so far. And digital agriculture, nowadays the trending term artificial intelligence and big data, where if you go to the meteorological department, you can get last 15, 30 or 50 years of data, a particular location where every year how much rainfall had happened, what are the sunny days, what are the rainy days, how much is the maximum temperature and minimum temperature. All data they can give for years together. That data can be inserted to artificial intelligence and they are able to predict this year how the climate will be, this year how much rains we are going to get. Some extent we can predict and accordingly we can plan. And mobile advisory services, nowadays we can find that government is sending the messages. If in the particular location any rain comes, they will send a message that you are going to get a rain today. So by avoiding the same manner, we can go for sending the text message to the farmer that in your location, go for this crop. In your location, go for application of this fertilizer or this organic manure, whatever. We can straight away send to the farmers. This is very important thing. Conservation tillage. How many of you know about the conservation tillage? What do? We don't disturb the soil much. Hope you know about the zero tillage and minimum tillage and all, right? So we don't disturb the soil much. Whatever the soil is there, wherever we want to plant the seed, there alone we make a hole and we place the seed and close it, so that when rain comes, soil erosion will not occur much. And cover crops, you can see the second one. Earlier I was talking about the soil erosion, but here if you see when we grow the cover crops, think about the soil erosion. But here if you see when we grow the cover crops, it not only covers the soil and also the rooting pattern of the cover crops will spread over all the soil and it will not allow the water to take away the soil particles. That's why in industrial we see the lawn. Whenever high rainfall occurs, through the lawn, along with the water, soil won't come out. And one more thing, organic amendments we can see, just you apply the organic manures. For example, let us say farm manure, or goat manure, sheep manure, or vermicompost, whatever the manures which are available at your surrounded area, you can straight away apply to the field, it will help you out. And efficient water management. As I was talking about the surface irrigation, try to shift from surface irrigation to the micro irrigation, that is drip irrigation. If you look at the drip irrigation, where laterals will straight away deliver the water to the root zone of the crop, and we can avoid growing of the weeds in the field. The reason is, wherever the plant is there, to that, as I was explaining of the site specific, same way here also, the water is straight away delivered to the root zone and surrounded area there will not be any moisture. When there is no moisture, obviously weed will not come. So that's the way how we can, and we can save the water up to 70%. See advantage. And here government is giving the subsidy to the farmers. Another advantage. And rainwater harvesting, even the government is insisting for the newly constructed buildings to go for rainwater harvesting, without which we cannot restore the groundwater table. We cannot recharge. So rainwater harvest is very, very important. And smart irrigation systems, now technology has come where from house itself, straight away farmer can turn on the motor to give the irrigation, and from there itself he can turn off the motor. So such a way the technology already emerged. And sustainable cropping system, crop rotation, that is nothing but instead of going for same crop, different crops as explained, and intercropping. So what are the crops which can fix the atmospheric nitrogen to the soil? Legume crops, very good. Actually, where they get nitrogen? From atmosphere. So though nitrogen content is, how much nitrogen content is there in the atmosphere? 78% of nitrogen is there, but it is not in the available form to the crop. So legume crops, they can fix the atmospheric nitrogen straight away to the soil, and it can help the crop which is growing near to it or in the next season. You know one more thing, legume crops can fix 40 kgs of nitrogen per year in the field, in a hectare. So that's the way how intercrop if you go, the main crop will be raised, and intercrop if you go for legume crops, yield you can get at the same time they can fix the atmospheric nitrogen in the field. And integrated pest management, just now applying the chemicals won't help us out completely, but go for the natural predators too. And livestock sustainability, improved feed efficiency. See, if you want to get the organic manure or if you want to get the poultry manure, straight away we have to depend on it, livestock. And if you don't feed it well, we cannot get it. So, improved feed efficiency also need to be worked out. Manual management, how much is supposed to be given to the crop, that much we have to provide. And animal welfare, even including the insurance of the animals need to be done for the livestock. And role of biotechnology, they are playing the crucial role. We should appreciate the biotechnology department where they are working on different departments including agriculture, where stress tolerant crops they are bringing up, where there is no moisture, how crop has to sustain. So it will never change. So we have to provide, it's our job to help them out. And farmer-centered innovations, indigenous knowledge, farmers are already having their own knowledge. And finally, when you go for Raweb to do the village program, you might be thinking that I can tell something to the farmers and I can make them to do well. No, in general, farmers will teach us much. Okay, participatory approaches, by conducting number of meetings to the farmers, we can help them out by giving the knowledge and capacity building. And here we see in sustainable agriculture, many of you might be thinking organic farming is there, natural farming is there, then why another term called sustainable agriculture? Okay, there is a difference between organic farming and sustainable agriculture. Sustainable agriculture is inclusive of all. We are not damaging it, and whatever we damage, everything we will repair it. That's the concept of sustainable agriculture. But when it comes to organic farming, to turn the inorganic farming land to organic farming, farmer takes 3 years. That is damn sure. If organic farming certification agent got a ZPNF with zero budget natural farming in Rayalaseema area, especially on orchard crops, where they are helping the farmers by giving different technologies and helping them how to prepare these natural products and all and applying. Like Jivamrit and Astras, these are the pesticides we can consider. And gender and youth inclusion, nowadays we can see the migration of youth from village to the urban areas, and empowering women farmers is important, and engaged youth in agriculture is also important. Let that, we can start, the days are coming where non-agriculture background students are coming to the agriculture and they are doing especially software. And sustainable value chain, reduced food loss is very, very important. If you see most of the hostels every day, how much food loss is happening. So we better reduce that so that we can look for the future generation. Fair markets. See now, middleman is increasing as days pass by. Whatever the produce they are buying from the farmer, when it goes to the market, it is even tripling. Okay, for example, they are buying papaya or watermelon 1 ton for 10,000 rupees. That means 1 kg 10 rupees. But when it comes to the market, 1 kg is around 30 to 40 rupees. It is tripling the price. Who is taking in between? The margin by middlemen. So we better avoid them, and straight away if we have the platform that will be helpful for the farmer. And traceability, whatever we done, everything every year we have to give the data to the farmer and follow so that it will help the farmers. And let us see the future pathways. Within 5 minutes I will conclude. So system-based approaches and innovative scaling and global collaboration. It is at the between the countries. If it is happening, that will be helping our farmers. Whatever the innovations that are done in our country, even other countries, let us exchange so that it will be beneficial to the farmers. Even global collaboration also will help. Research and education, interdisciplinary research, not only within agriculture, just as I said that between the biotechnology, agriculture, microbiology, and food science and technology, any other department, if we have the interdisciplinary research, that will help us out. And knowledge transfer, whatever we innovating, that we better transfer one another. Even AARC is helping a lot in agriculture. Extension services, whatever the innovation is done, let it not remain in the lab itself, let it go to the farmer's field. And role of international cooperation, knowledge sharing aside, funding mechanism. So some of the organizations, they are providing the funds like Bill and Gates and other organizations also are there, they will be providing, and technology transfer. Measuring sustainability, indicators and metrics. Every year, how much really it is went to the land. That measuring should be done. Then only we can see the progress. Life cycle assessment. That means when we really adopt the sustainable agriculture, how much really the people's lifestyle has changed. You can observe in between you that nowadays when people are going to the market, they are preferring the organic products. So slowly lifestyle is changing. They are not preferring much to the inorganic. Though the price is high, still they are preferring. And monitoring and evolution, these all need to be monitored and certified, and agencies should be, then only that, okay, everything will be done in straight way. And out of my presentation, the key takeaways are sustainable agriculture is not an option, but it is essential in our individual life, and no single solution for all contexts. Whatever the solutions I gave, if I adopt one solution, will it solve our problem? No. All need to be worked out. Then only problem will be solved. And innovation must be inclusive, ethical, and ecologically grounded. Farmers are agents of change, not just beneficiaries. The future of agriculture determines the future of humanity. My conclusion, the challenges of the 21st century is not only to feed the world, but to do so without consuming the future. Thank you. Any questions?
Thank you.
A very good afternoon to everyone. Here I am going to explain about the title Evaluation of Post-Operative Causes and Mobility Scores in TKA patient with FNB versus ACB with a prospective observational cohort study. Actually, we are going to assess which block is working better. We are going to assess which block is going to prevent the pain. Here we are mentioning the two blocks. One is femoral nerve block and another one is adductor canal block. These blocks are given after TKA surgery to prevent the post-operative pain. Actually, the TKR is a very painful procedure to prevent the post-operative pain. The femoral nerve block and adductor canal block, the both blocks are given. The total knee arthroplasty is commonly called as a surgical procedure where we can replace the damaged knee joint with the artificial one to relieve from the pain. And to prevent the post-operative pain, we are giving femoral nerve block and another one is adductor canal block. And the effective post-operative anesthesia is crucial for the early mobilization and whereas for functional recovery and to prevent the complication. There are two types of anesthesia, one is femoral nerve block and another is adductor canal block. And whereas the early mobilization is critical to preventing the deep vein thrombosis. Deep vein thrombosis is a condition where there is a clots occur in the near the legs region. And if we leave that condition, it may also lead to the pulmonary thromboembolism. Next, diagrammatic representation of femoral nerve block and the adductor canal block. Femoral nerve block is the main nerve which carries the sensory and motor signals to the thigh, whereas the adductor canal block can be given to the surface nerve, which is the sub-branch of the femoral nerve block. Whereas in the femoral nerve block condition, the entire motor signal and sensory signal will be blocked. In the adductor canal block, only the sensory will be blocked. There is no motor signal will not be blocked. So most of them prefer adductor canal block. So the study objectives are to evaluate and compare the impact of ACB and FNB. Safety. The safety is assessed by using the Morris fall scale. It is going to assess how often the patient may have a risk of falling after the surgery, and functionality. Functionality is going to assess by using the time to upward test. In this test, where the patient can how much time he can take to stand or sit in a chair or else turn a back. How much time he can take to stand. Next, in the motor strength, in this the quadricep grading is used in the medical research council. By using the scale, we can calculate the motor strength of the patient. And analgesia is the pain intensity is measured by using the visual analog scale. Next methodology, we are taking the 50 patients who have undergone unilateral total knee arthroplasty, and we will be dividing into two groups. Group A, Group B contains 25 and 25%, and assessment is conducted at 24 hours to 48 hours post-operatively. And we will result fall risk must take the highest score indicates that the patient is having more fall risk, whereas the lower score indicates the patient is having low fall risk, whereas the ACB having only 32.4% score, whereas the FNB is having 40. This indicates the FNB group is having high risk of falling.
So if the blood pressure is not enough, the producer is skewed. Why should we do
How can we increase the data? We have collected only 50 questions. Sure is your provided separate. But when it comes to the diagram of the scores, the data tells us we have collected data. What is the meaning? For the 52. What is the physical meaning? Actually, we will prepare a questionnaire. Into the questionnaire, we can ask the patient based on the questionnaire, we will give the score, and that scores we will calculate. So why 32 is like that? What is it? That is the score. Is it low score, average score? Yes, that is low score compared to the moderate risk. So low score is there, mean score is yes or no? The level is not there, no risk. We are calling moderate risk. Not exactly. Isn't it positive? Yes or no? Then what did you prepare? What did you do there? Simply reading from the slides. Does it make sense? Okay. Last slide. This is the graphical representation. The summary, ACB offers comparable analgesia to FMB while drastically improving the patient safety. 88% of mortality preservation, significant reduction of TUG time and Morris quality. And final recommendation, ACB should be preferred as a regional anesthesia technique to enhance the functional recovery of the patient in safety.
Where tele and ICU, the combination tele means communication and ICU comes with the healthcare sector, where how integrated in order to optimize the patient healthcare and to save the patient. So the background where tele ICU is an advanced equipment where the communication with the patient and with the nurses with the patient's data and with the monitoring center, where we will be where telecommunication technologies with real-time data integration, it connects with the nurses, intervenists and doctors, where the ICU is deemed to be with the speaker and all, and monitoring with the ECG data of the patient. So the main objectives of the tele-ICU was to evaluate the risk of tele-ICU enabled hemodynamic alert verification in order to save the
I need printer of silence from the conference hall. Please.
So the primary objective of this study was to evaluate the outcomes hemodynamic alert verification, and the secondary outcomes of the study was to how fast the patient, how fast the nurses and the intervenists and doctors react to the dynamic hemodynamic events and give the patient the necessary assistance. Where to compare the IHCA. IHCA stands for in-hospital cardiac arrest. How much secondary objective of the study was how to minimize the IHCA in the hospital settings, and how to decrease the length of hospital stay, and how to discharge the patient as soon as possible. So the materials and methods, the study design was the prospective multi-central observational study from the September 2024 to August 2025. it is done with the 3 hospitals in association with the Aster Ramesh, Ongole, Guntur, and as well as Vijayawada. So study population was 5,253 patients with ICU wards and ED continuously monitored with the greater than 2 hours of duration in their admission to the hospitals. So the exclusion criteria are orders and the monitoring below the 2 hours we have excluded, and any discharge or transfer or pacemaker or pregnancy population were excluded in the studies. Tele-ICU monitoring system, we have taken data with heart rate, BP, SpO2, ECG and respiratory rate with automated alerts. Alerts were verified by the tele-ICU monitoring intervenists, those are nurse or other healthcare professionals with real-time communication with the hospital. So the data collection we have taken demographics, diagnosis, comorbidities, Apache score, it is a score where we use it to analyze the patient's score in the ICU for the mortality rate, interventions and LOS length of hospital stay, and mortality recorded in the tele-ICU databases. The outcomes, primary was to in-hospital mortality and the secondary was diagnostic accuracy, how well the intervenists will be assisting to the doctors or as the concerned patient in order to minimize the risk that he has faced at that time. So this is the first picture was the Ramesh Hospital monitoring. You can see the grey color that is the speaker, and the second picture was the tele-ICU monitoring. Intravenists will be monitoring 24/7 in the tele-ICU in order to give the patient, in order to minimize the risk of the patient.
Is it their system or your system?
Their system only, sir. They are using it.
And what is your input?
We have done this study, sir, in the Aster Ramesh hospitals. In the Ramesh hospitals, we have did this study, sir.
You just took the data and analyzed.
I was part of the study as well, sir. I was in the study as well.
So, if it is your PhD work,
Sir.
what is the in your input?
I mean, what I did from this study you are asking, sir.
PhD means doctors only.
No, sir, it is
research.
Research article, sir.
Only for the analysis.
No, sir, we have minimized the patient mortality rate by this, sir. Yes, sir. Yeah, sure, sir. Yeah, okay. So here we can see, sir, comparison chart where pre-tele-ICU before this study, out of 1,000 admissions, we have a mortality rate of 8.2. Post tele-ICU, this will be, it is concised to 4.3. That means almost 47% of the 47% has been decreased post tele-ICU, sir.
What is tele-ICU?
Tele-ICU means nothing but if a patient admitted to the ICU ward, we will be monitoring his ECG, vitals, heart rate, everything, sir, like A to Z from the other.
So the same patient come again.
Sir.
Number is increased.
What number?
The same patient comes again
to the ICU. Okay.
Tele-ICU number increases.
No, sir, it is real-time data monitoring. If, for example, an ex-patient has admitted into the hospital, we will be monitoring his vitals, everything, ECG, A to Z, the parameters in the tele-ICU, sir. Once he discharges, his data will be stored. Once he again, if X has admitted again, the data will be again monitored. It is nothing but a tracker of the patient. Whenever he admits. Sir?
Will not increase the overall data by one.
I mean, this data was about 1,000 admissions, sir.
So these 5323 are unique data, unique patients.
Yeah, unique patients, exactly, sir. Unique patients. Yeah, and the median age of the patients was 58, sir. The median age for the patients, and the criteria was more than 2 hours and pregnancy, so that is an exclusion.
Somehow I am still not understanding what is your input there in the data.
In this study, sir, we have minimized the mortality rate of the, like, swift action from the nurses and staff, sir. Like if 13 beds has been installed in the ICU, sir, 13 patients will be there, and then 2 or 3 doctors will be monitoring them. So 3 doctors monitoring 13 patients or 14 patients, that is highly impossible. Even the nurses, because the procedures will be going on simultaneously. So the tele-ICU intervenes. So tele-ICU intervenes, they will be having their ECG data, everything for a separate patient, sir. They will be monitoring them. If any high risk or else the irregular heart beating or any tachycardia situations, they will be alerting the doctors, sir. So by that the doctors will be alerted in the Ramesh hospital and they will be intervening, sir. So by this study, I mean, concluding this means 38 deaths we have been saved throughout this study, sir. Like 38 patients were alive and now they are alive. 38 people. So and for 1000 admissions, if we take that as an data, so from 8.2 to 4.3, the mortality has been decreased, sir. So it is an outcome summary where alerts detected was 57%, mortality 15%. Median time response was 16%. The total patients were 11. So a total 5,253 adult patients were continuously monitored during the study period. Among 64% of males, and the median age group was 58 years. Clinically significant hemodynamic alerts were detected in 2,278 patients, that is 43.3, and 456 patients, 20% of the output required immediate clinical interventions. So we have immediately alert group was 456 patients, sir. By this we have alerted immediately. So 20% of the group was elected immediately, sir. And the most frequently observed hemodynamic event was tachycardia, sir. As I mentioned earlier, tachycardia was detected and the doctors and nurses was alerted swiftly, followed by bradycardia, sir, 26%, hypoxia and hypotension, sir, with their respective percentages, where the hypertension accounted for only 1% of the events. Most events occurred in the ICU settings, sir. As we discussed earlier, only the ICU patients will be monitored by the help of tele-ICU systems, reflecting the higher severity of illness in these patients. The tele-ICU alert system demonstrated good diagnostic performance with a sensitivity of 79.2 and specificity of 80.1%. The positive predictive value was 68.7, negative was 87.1. The median time alert generation intervention was just 12 minutes. Okay, sir. So finally, the patient out of, as I said earlier, from 1000 admissions, from 8.2 to 4.3 has been decreased and representing an approximate 47% reduction, and an estimated 38 cardiac arrests were prevented during this study. Thank you.
My topic is smart farming and the human factor cultural barriers to adoption. paper, I would like to show what are the barriers we are facing in the farming sector. My name is G. Pooja. I am a research scholar from Jayati Vidyapith Women's University, Department of Management and Commerce. Actually, the farming and other sectors are maybe majorly, you may think that it is not related to the management, but management plays a role in all aspects in our daily lives. So my major paper, this paper majorly concerns with the culture, why it is a major role in farming. Can anyone answer why culture is important in farming? Because culture is the important factor, how the farmers adopt to the soil texture and other factors. So, and the atmospheric reasons and all the criteria are the major impacting factors to be treated. So, that is why I am participating. So this paper, the cultural aspects, the gap, and how it can be treated is being seen. So why this study is needed? So cultural barriers, trust, identity, global evidence, I am also showing it. The cultural barriers may be gender norms, symbolic meanings, and other all factors, where the modern farmers like adopting to the IoT, Internet of Things, or AI droning systems and climatic technologies being adopted. But there is a gap that needs to be taken into the consideration, like trust and identity. How these technologies can create a trust in the farmers, how they can be identified by the farmers into adopting the technologies. Global evidence, India, Africa and South Asia, because these areas, the soil texture is different from place to place. That is why these studies have been taken. The objectives to finding this paper is to examine how adoption outcomes are influenced and how generational differences, symbolic meaning and cultural identities affect the farmers' choices, and explain the adoption factors, patterns, diffusion of innovations, frameworks and social technical system theories. So the theoretical factors that I have taken for this paper is, first is the technology acceptance model. Why the technology is accepted by the farmers being because they face major backlogs, like if there are floods, they are unable to bear the consequences and they are unable to face the consequences and how it can be treated. Diffusion of innovation is a theory that influences the relative advantages like technology, traditional values, socio-technical system theory and intersectional. So the major research gap here, we can see the overemphasis of technical efficiency. If farmers totally depend on technology, how they can technology that they will give 100% productivity or 90% productivity to the farmers? They cannot make it because the soil temperature and the soil, what kind of soil it is, they can identify, but they cannot measure what the soil has in it. Limited integration of socio-cultural dimensions. Like this shows that evidence from Africa and India and also from the Andhra Pradesh like Kurnool and other districts, it shows that the major shape adoption, like what farmers are using in the farming. They may vary from one kind of soil and what type of crop they are going to make. And neglect of equity and intersectionality. Like there is case of mechanism, as we have seen, like they cannot adopt to the technology and they cannot implement in a area of small place. They have to be taken at the larger scope. So this paper methodology relies totally on the secondary data of case studies and how the theories are efficient and they are made practical to the.
Where did you publish this?
I have not published this.
But, so should you write the lines as such? With the PowerPoint.
Like sir?
Is it the paper or PowerPoint?
No sir, this is the PowerPoint presentation.
So you should not write all this, no?
Yes sir, but for the audience, I have an
Audience, you should tell.
Yes sir, that is why I am not reading.
Brief points required.
Yes sir, but for some audience it is majorly they focus on studying the content, that is why I have placed this content.
That is their trouble.
Yes sir, but we have to consider the audience, that is why I have to.
That's not your trouble, you are not teaching here.
Yes sir, but it has to be taken to the audience. They have to know.
And why did you explain the references? Some people may actually like to read reference also.
I have explained the major theories I have taken for
So write 3 points here. What is your main objective?
My main objective is to find what type of culture and the technology barrier in the farming sector.
So that's a review idea.
Yes sir.
What are you saying, research design?
Research design is how I have implemented, I have made these papers.
If it's a review paper, it's somebody else data. You are just telling it. Research design means you are not done.
Yes sir, I have done that.
Review paper, research paper.
It's review and research because this was like because the culture aspect has been not considered till now. I have googled and I have searched many of the journals and there is no single topic that concerns with the farming culture that shapes the adoption of technology. There is no because there is a barrier that people are concerned with adopting technology, but they are not concerned with how the process or how the farming should be taken and how the farmers are doing it.
This is one perspective. Your opinion.
Yes, sir.
Your opinion is review. It is review only.
Yes, sir.
So no research design. It is a strategy.
No.
Strategy of doing this. We carry on. Last slide.
This is the future research directions I would like to. Empirical validations that trust, risk and cultural fit need to be taken, and participatory action is because I have not done any on-site survey or research any other things because there lacks some of the implementation of theories so that the practical sense will be valid. Thank you.
are coming to the final session of the valedictory function. Please wait 30 minutes. 4 o'clock వదిలేస్తాము. And please tell your feedbacks regarding this seminar. మీరు ఫీడ్బ్యాక్స్ చెప్పేవాళ్ళు డిఫరెంట్ కాలేజెస్ నుంచి వచ్చారు కదా, యూ జస్ట్ కమ్ ఫార్వర్డ్. ఎందుకంటే మనకి చీఫ్ గెస్ట్ రావడానికి ఒక 10 minutes పడుతుంది. సో ఇన్ ద మీన్ వైల్ జస్ట్ టెల్ యువర్ ఒపీనియన్ అబౌట్ దిస్ కాన్ఫరెన్స్. ఐఎమ్ ఎంకరేజింగ్ outside students, please come forward. There are the students from KLU, SRM. Now please make pin drop silence. The students from other colleges, please come forward and tell your feedback. Randi Dixi, లేకపోతే కొట్టు కొట్టుగా అన్ని తీసుకొస్తాం. రావాలి Dixi. Come. Proper వెళ్ళండి. మా 2 days చక్కగా సైలెంట్గా ఉన్నారు. ఒక ½ an hour మీరు స్పెండ్ చేస్తే మిమ్మల్ని మేము కొంచెం పంపించేస్తాం. అర్థమైందా? విండో సైలెన్స్గా ఉంటుంది. కమ్.
I am from Geetham University, Vizag. First of all, thank you for all attending this, and my sincere thanks to all the organizers and department for each, and special thanks for the sir from Adapter Microbiology department. Sorry for not knowing. From the starting day, I was very nervous to attend this because it is international conference. I didn't know this was my first interaction session, so I am unable to interact. By the starting stage, I saw the father of this Loyola College. They received us very well, and it was a pleasant environment, and the staff was very friendly with us. They treat very well, and finally I am concluding that I am very happy to involve in this session and present, and I hope I will soon see you again. Thank you.
Very much thank you to Sohail for your wonderful speech. Thank you.
Good evening, everyone. Here is Veera Magina from ICFLET University. From starting receiving hospitality to this valedictory function, it's adorable excellence, sir. I thank heartful to Andhra Loyola College for conducting their international conferences and welcoming us. Thank you, sir.
Very good evening to one and all who are present here. You are very lucky to have the international conference, and every year we are going to present these conferences. And myself, I am Prasanthi, full-time research scholar from Yogavemna University, Kadapa, and this is a better opportunity to present our papers as well as the research work which we have done. And as a students, it is your lucky chance to share your poster presentations as well as the knowledge which you have learned in your classrooms to the presentations. So this is the better opportunity which I think, and moreover, I am the alumni of this college. I studied my undergraduation, post-graduation here itself only. I'm happy being the part of, as a research student from the other university here. Thank you so much for giving me this opportunity. And this today international conference was done very well by the conveners. It is a good luck, sir. Thank you so much, sir.
Please come forward from KLU University, SRM. KLU University. You? KLU.
today's international conference was with the speakers of multidisciplinary of zoology, from chief scientist from CSAR and all. Not only this session we have, I also have commented in LinkedIn. Sir shared, Shanbaja sir shared one post regarding it. Like it's very our pleasure to be here. And also not only that, we are these conferences are especially for sharing of knowledge and also for networking. And here I have seen maybe from Allareddy College and CSIR, and also Rafi sir also there here. We met in 2021 sir, 2021 actually. And also Nadine sir, it's very nice to. And also I am thinking to come again if there is a chance in National College. Thank you.
Now please, SRM, University, KLU. Anybody from Loyola College? Among from our college, from different departments, please come forward and tell your opinion. Loyola College was our leader. Mockler ఉన్నారా ఇక్కడ? మొక్కలు ఒక లెఫ్ట్ ఎవరు లేదన్నమాట. థ్యాంక్ యూ. మొక్కలు ఎవరు లేదు మా కాలేజ్. Anybody would please come forward from our college? Girls, biotechnology, microbiology. I am requesting any one of the resource persons, please share your please share your experience.
To start with, the 2 days have been very phenomenal. I like the hospitality there for the different people, especially the students. They have been very excited about all this. And one thing which was really disturbing was the audience. Other than that, everything was super good. The food, the tea, everything was great. Even the faculty members, they have been outstanding in the part. Marshals are these people. Very excited, very energetic part. So we are going to meet different people. Today morning only I did an advance booking for next year. Next year whenever the conference is there, please give me the letter first. I did an advance booking even in the ESG hostel. I lived in the ESG hostel. Food was super. Chala bagundi. Very super. So I did an advance booking for next year. So that was super. So I called him the Mahesh Babu.
In biotechnology department. So I enjoyed very nice, super experience it is. Thank you so much.
Good afternoon. I'm glad
to be here. And thank you for the invitation, especially to Dr. Jemima, the head of the. See, I would like to say one thing to all the students, irrespective of the department. When I was studying my undergraduate course, in my university there will be flexes, the standings will be there. There is national conference, there is international conference, there is workshop, but knowing not anything about all those, not at all. We have no awareness about it. But when we are entering to final year or MSc, then we realize, okay, those certificates are helpful it seems. So let us attend, let us participate so that we get the participation certificate. So you all are saying, right? We'll get the participation certificate, so we are attending the conference. And one more thing, when different departments are there and when they are presenting the lead paper or the keynote speaker or the guest paper, whatever it may be, whatever related to us, some interest we will be having, and whatever not related to us, no way we find the interest within us, right? And something, though they are speaking English, but nowhere it is connecting to us, something other language. But you see, my experience is when you are attending the conferences, damn sure you will be getting some knowledge out of the conference. Above all, if you are really interested, you will be getting the new connections with the delegates who are coming out from different industries or any research organization, whatever. If you are really interested, they may be the pathway for you to get the job too. And also, if you are interested in research work, they will help you out. And my thing is, I started attending the conferences. Later, I started presenting the papers. My small work might be very small work. Let us start presenting. Then I realized that okay, there is a best oral presentation award. There is a best poster presentation award. Why don't I try? And when I tried, first time I didn't get. We may not get, but presentation is more important, so that we can improve our presentation skills. Then competitive spirit will be growing within us. Then by this, I got I think 5 best oral presentation awards. Nothing to be honored, but as we are trying and attending more and more, for sure you will be getting the. Today I didn't present any of my research work, because most of you are students. If I present my research work, something which is not at all related to you and nowhere you are interested. But my sessions today is different. I didn't like what I can say. Present my PhD work even. Even I said that when I was presenting. So, but go on attending the conferences or workshops and get the certificate. That is very good thing. But above all, get the connections and present the papers. Small work, review paper. That is fine. You may not doing the research work, but try with the review paper. So whatever the research work done in your interested area, all the research work you collect it and present. That will be fine. And next conference you start doing your own work, then present it. That's the way how you have to improve. Nowadays the competition is too high. Once you go to the market, when you go for the small job, behind you will be finding the hundreds of job seekers. So to compete all those people, it's very difficult unless you develop your skills. That's what I can say in this conference. Thank you so much for this time and all the best for your future. Yes, ma'am. My PhD paper is resource technology in maize followed by the rice, where I have used the organic amendments like, how many of you know water hyacinth? The plant, Telugu burrapute can't. So I use that plant because it is, when we go to the farmer's field, we may not get enough organic manure like cow dung or whatever, but burrapute is enough. All is about we have to take it out and dry it and apply to the field. So it is free of cost, man. So I worked on it and applied to the field. To conserve the water, if you observe the water hyacinth plant, it is having the spongy-like tissues in the paper. It will absorb the water out. So it will keep the moist to the soil. And apart from that, I applied the sugarcane trash. I checked out which is working well. On field directly, not in the lab. On field directly I have worked out. Okay, that is brief. I am telling you, not in depth. Ma'am. Currently I am working at the Mallareddy University, Hyderabad, but where I study is Annamali University. Ma'am? botany. Yes ma'am, I'll let you know. You can ask ma'am, she'll give my details and we can work together if you are really interested. Actually, we are working for PG and PhD also. Currently, already I guided one student and right now, under me 3 students are there, PG students and 1 PhD student. We are working on things. Okay, thank you.
Good evening. I am a research scholar from Yale University. First, when I heard about this, the last moment on the last day, I thought it is very good to attend this kind of multidisciplinary things. So I joined at the last moment and I prepared my oral presentation. But when I came here, I was so much gained so much of knowledge because of the great presentations, and I'm thankful to be here, and I'm thankful to present whatever I have presented. Thanks.
Experiences from our college faculty, from department faculty, minimum. Dr. Ramesh sir. Now I invite Dr. D. Shokumari Madam, Vice Principal from the college, just share the experiences. Madam is so, Madam conducted many seminars, workshops, nearly for these years, 100 seminars were conducted by Madam. Madam, please share your experience.
Good evening, everyone. Good evening, students. This conference is for you only. We experienced with young number of programs. We organized, we explored, and gained lot of knowledge through experience and participation. So, participation is very, very important, even though it is not possible for you to present a paper. After attending this program, I am expecting all the AMC students and also participants from the other colleges, degree college level. It's a kind of ongoing process. Life science means it's a continuous process. Every day there is a new innovation, new change, new discovery. Tomorrow is new, today, so yesterday is past, and today is unknown. So, always ready to accept new innovations and also involve in all the research activities so that we explore your knowledge in all the fields. My experience in this college is, because of this college only, I learned many things. Before I came to this college, it means almost 1998, I don't know about what is computer. But today, I prepared a digital herbarium for AMC Flora, a website for Department of Botany, and blog for Department of Botany. From last 14 years, continuously, everyday program we are maintaining, we are uploading in the blog. This is what I learned from this college, because discipline is our primary motto in our college. Every day I followed only the discipline. If I have that activity, first college in Telugu States, DBT Star College scheme, 70 lakhs fund we received from the Department of Biotechnology, Central Government of India, and the coordinator of that. That is the first one. And I received UGC major project, 14 lakhs from the New Delhi Central Government. Again, that is the major project I received from the Central Government. And 2 minor projects from Botanical Survey of India and National Botanical Garden and State Botanical Garden. Total 5 grants I received from the central government and state government. Ours is the only department, I am proudly saying, botany department is the only department in the college received total 1 crore projects from the government. And another achievement, and our tag and another department, botany tag, we published 3 volumes of Flora of ALC. The 3rd volume consists of 1,000 plant species belonging to Andhra Loyola College. So, everything I gained from the college, almost all the projects based on biodiversity, and that biodiversity is also belonging to ALC. That means my A to Z achievements goes to ALC. That is my opinion. So, 2016, last night I remember regarding this conference. 2016, in the month of September, I think September 17th and 18th and 19th, or 19th, 20th and 21st of December, if I am correct. Yes, before December Christmas holidays. First time in the college, the Department of Botany organized international conference with 21 speakers from different universities, different states, and 3 members from the states. 3 days continuously we organized international conference. That is the first conference in the college. So, again, that is the first achievement towards the life sciences. I am using the word life sciences, especially the Department of Botany. Another landmark in the Department of Botany, I received already, I told you, more or less 1 crore rupees from the government based on interest. But I have interest. If I have doubt, I will ask the first year student how to go. So I have no limitations to learn the things either from my age group or below age group. If you want to achieve the things in your life, always maintain the things from the bottom of the bottom. You don't think about you are the great. Number of developments are there. What you learn is 0.1%. Every day you are a student. If you have that concept in your mind, a greatness and a proudness and I received 3 important awards, 4 important awards in my life. With 90%, 89.90% in the field, 89.9% in the degree. Best outgoing student in botany in degree. Gold medal in PG. Best state research award from Government of Andhra Pradesh. Best research award from Commissionerate of College Education. State Best Teacher Award from Government of Andhra Pradesh. ఇవన్నీ నేను ఎందుకు చెప్తున్నాను అంటే బికాస్ ఐ స్పెండ్ లాట్ ఆఫ్ టైం హియర్. నన్ను కొంతమంది తిట్టేవాళ్ళు, ఏం తేడా కదా 7 వరకు కాలేజీలో ఉంటారు, 6 వరకు ఉంటారు. బట్ దట్ ఇస్ మై ప్రాబ్లం. మూవింగ్ ఇన్ ద గ్రౌండ్, speaking along with the plants. Just I want to say every day, A to Z. That is the observation every student need to have. If you want to go for research, if you don't have patience, if you don't have observation skills, if you don't have idea to listen and patience to listen, that profession is not good for you. Till 1998, I don't know anything. Once I entered into the college, I observed the dedication of the fathers. They are the motivators for me. Every father motivated me, do this, write this project, do this conference. Almost nearly 18 national conferences, 8 workshops, 1 international conference. I am the organizing secretary of the different programs. But even today also I learned something from Arvind sir. What I learned last night, I observed. What I have learned from him as a convener. Please remember, every human being has his own talent. ప్రతి మనిషిలో కూడాను ఒక టాలెంట్ ఉంటుంది. ఎవరైతే ఆ టాలెంట్ అబ్జర్వ్ చేసి వాళ్ళలో ఉన్నటువంటి ఆ స్కిల్ తీసుకుంటారో, ఆ స్కిల్ని అడాప్ట్ చేసుకుంటారో, దట్ ఈస్ ద ఫస్ట్ స్టేజ్ సక్సెస్ ఇన్ అవర్ లైఫ్. వాడేముంది అసలు అదేముంది, అది కూడా ఒక పెద్ద ఎక్స్పెరిమెంటా, స్టార్ట్ ఇంట్రెస్టెడ్ టు లిజన్ ఇన్ ఇంగ్లీష్. ఓకే, ఎవ్రీ డే వీ నీడ్ టు థింక్ అబౌట్ వాట్ ఐ లర్న్ డే టైమ్. And also, every student has his own knowledge. Just observe the skill in the skill and identify the skill. I want to thank the management of Andhra Raila College in one aspect, in a personal way, because from last June, I am closely related to department. Every day I will sit in the lab and I call the students, I engage some program, I organize something. But now, the management appointed me as vice principal of first year English students. Every day I want to see 1320 students personally. I want to trace the talent in each student. Now that is my target. Subject wise, I am not great, but I am satisfied. Now what is my target? I want to see the skills in the students. There are different types of skills students. Please remember, just and 1 hour, wasting 1 hour time is very easy. But in between 2 o'clock till 3:30, nearly I spent 1 hour with 3 students. 1.5 hour with 3 students. Number of things I explained here. And one parent just told me they came. He used one word. ఆ పేపర్ మీద రాసేటాడు, నా కళ్ళ దృష్టిలో మేడం ఫస్ట్ టైం ఐ వాజ్ సీయింగ్ a woman with heart, a woman who is dedicated to student work. This is the word they know. That is enough for me. Research scholars, eminent speakers are there. They are greater than me, but professional satisfaction always comes when you are experienced. Please remember this. Even though I am getting 3 lakhs of salary at present, think about that. Every day I experience the results of the students, the behavior of the students, the parents' information about the students, their marks, their good behavior, and how I want to motivate them. How can I motivate the students? Every day this is the target. Here each student, you need to have a target. గాలిలో దీపాలు అంటారు. యు ఆర్ నాట్ గాలిలో దీపాలు. గాలిలో దీపం అంటే ఏంటి? గాలి రాగానే ఆరిపోతుంది. That means without target in the life, if you don't have target in life, nothing will achieve. If you want to achieve something, today onwards, you work hard. Really, yesterday onwards, a lot of pain in my heart. 50% of the students spending their time outside. Valuable time, quality of time. Even number of research persons are there. They are giving lot of information, where to go, how to reach. But this is the age. Generally, చిన్నపిల్లలు కాబట్టి you want to spend some time, okay, leisurely. But ఇటువంటి కాన్ఫరెన్స్ కండక్ట్ చేయాలంటే 1 month డే అండ్ నైట్ వర్క్ చేస్తారు ఆర్గనైజర్స్. బట్ 1 day మీరు కూర్చోవడానికి మీరు కష్టపడుతున్నారు. దట్ మీన్స్ యూ నీడ్ టు లెర్న్ ఫస్ట్ హౌ టు గెయిన్ పేషెన్స్. దట్ ఇస్ ద ఫస్ట్ పాయింట్. హౌ టు అచీవ్ టార్గెట్. అండ్ ఫైనల్లీ how to gain your dream. అందరికీ డ్రీమ్స్ ఉన్నాయా? Yes or no? We have dreams. డ్రీమ్స్ ఉన్న వాళ్ళందరూ చేతులు ఎత్తండి. Raise your hands. ఇప్పుడు కూడా చేతులు లేకపోవట్లేదు అంటే కమిట్మెంట్ ఇన్ యువర్ లైఫ్. కూర్చున్నారు ఎలా అబ్బాయిలు? నో. రైట్ బాయ్స్, I think you learned a lot from this conference. That is goal setting. Setting a goal is the target of this conference. So, ఎంతమంది మీరు goal set చేసుకున్నారు? Raise your hands. No goal setting. Very good. The participants from other colleges, they raised their hands. But anyhow, I am happy with my profession because my concentration, my focus is on my profession. If you have focus on your target, your goal and your target, definitely you will success in your life. These are the words. Thank you so much for
శివకుమారి మేడం, వైస్ ప్రిన్సిపల్. సో మేము మేడం నుంచి చాలా నేర్చుకున్నాం. యాక్చువల్గా నేను చాలా చిన్నపిల్లడిగా ఇక్కడ వచ్చాను. మేడం నాకు మేడంతో ఉన్న ఇంటరాక్షన్ ఏంటంటే నాకు చాలా ఎక్కువ కోపం ఉండేది ఫస్ట్లో. మేడం ఏమనేవాడు అంటే బాలచంద్ర కోపం తగ్గించుకో అనేవాడు. బట్ అప్పుడు చెప్పినప్పుడు ఎందుకంటే నాకు ఒక మదర్ లాగా రోజ్మెరీ మేడం అంటే నాకు డైరెక్ట్ టీచరు. నేను రోజ్మెరీ మేడం దగ్గర నేర్చుకున్నాను. శివకుమారి మేడం దగ్గర లైఫ్ అంటే ఏంటో నేర్చుకున్నాను. లైఫ్ ఎలా ఉండాలి, కాలేజ్ ఎలా ఉండాలి, ఒక మదర్ లాగా, అంటే ఇంట్లో మదర్ కాదు, ఒక మదర్ లాగా మమ్మల్ని ఎప్పుడు కోపం వచ్చినా నేను మేడం మీరు ఎందుకు మేడం మమ్మల్ని అలా తిడతారు ప్రతిసారి అనేవాడిని. పాప ఆ టైంలో మేడం తిట్టినా ఏదన్నా తీసుకుంటే ఆ టైం కోపం వస్తుంది. తర్వాత ఆలోచిస్తే అని చెప్పింది కరెక్టే కదా. అందుకనే స్టూడెంట్స్ మీరు కూడా ఇప్పుడు స్టూడెంట్ లైఫ్లో ఉన్నారు కాబట్టి కొంచెం మోరల్ వాల్యూస్ అవి ఉంటే చక్కగా మీరు ఇప్పుడు ఉన్నాడంతా మంచి పిల్లలు. నో డౌట్ అబౌట్ దట్. ఏదైనా బయాలజికల్ సైన్సెస్ పిల్లలకి చక్కగా సాఫ్ట్ టీచర్ ఉంటుంది అంటారు. అంటే హార్ట్ కోర్ బ్రెయిన్ కాదు మనకి. ఈవెన్ మనందరం ఒకే కోట్లో ఉన్నాం. ఓకే, సో ఇవన్నీ నేర్చుకోండి. రీసెర్చ్ చేసుకుంటే, పిహెచ్డి చేసుకుంటే ఇట్స్ వెరీ యూస్ఫుల్ ఫర్ ఆల్ ఆఫ్ యూ. అండ్ మార్నింగ్ మనకు I-20 ఫీవర్ వాళ్ళు వచ్చారు, అబ్రాడ్ కన్సల్టెన్సీ. వాళ్ళు నియర్లీ 35,000 మెంబర్స్కి వాళ్ళు అబ్రాడ్ ఆపర్చునిటీస్ అంటే లైక్ ఎంఎస్ ప్రోగ్రామ్స్ అవన్నీ రెడీ చేసి పంపించారు. ఓకే, అండ్ వాళ్ళని మీరు కన్సల్ట్ అవ్వండి. ఆఫ్టర్ పిహెచ్డి, ఆఫ్టర్ పీజీ తర్వాత. ఓకే, థ్యాంక్ యూ.
Good evening, everyone. Myself, Dhananjay. I
only microbiology, biotech, food technology different. Other different sales. So recently, 12 months back, I joined as a faculty in Department of Microbiology. So right now I'm working under the HOD, Bharat. And this is my first international conference as a coordinator for this one, as a member. But earlier I heard it from my department in Geetham University, but this is the first time I involved as a professional. And I enjoyed a lot participating in all these activities. And along with that, I enjoyed with you guys in the class, along with this evening. But in my concern, I have seen that some people, chala work endante, you didn't recognize that this is international conference. This is not a fest. This is international conference. That means here, the sharing of the knowledge among the people. Okay, ఇక్కడ ఒక knowledge ఒక share చేసుకోవడానికి వచ్చాం, not for the enjoyment purpose. So, if you want to go for international conference, మీరు ఒక బయట ఎక్కడ international conference వెళ్ళాలనుకుంటే, almost they charge about 2,000, 3,000. But along with the travel expense, everything you think. But you have given a nice opportunity. మీరు ఫ్రీగా ఇంకా ప్రొవైడ్ చేశారు. But only less people are participated. Only very less people are participating. But I thought that I'll get a more abstract than expected. But I got abstracts అయితే వచ్చాయి, but participation అయితే తగ్గింది. You have very good opportunity. ఇంత తక్కువ అసలు without fees you are getting an opportunity. You can utilize it. మీరు యూటిలైజ్ చేసుకోవచ్చు. ఎందుకంటే అదే బయటకు వెళ్ళి కొత్త నాలెడ్జ్ నేర్చుకోవాలన్నా, కొత్త కాన్సెప్ట్స్ తెలుసుకోవాలన్నా యూ గెట్ సమ్ టైం. ఎందుకంటే ఆ నేచర్కి అడాప్ట్ అవ్వాలి అండ్ ఆ పొజిషన్ తెలుసుకోవాలి. బట్ యూ హ్యావ్ ఆపర్చునిటీ. ఎవరో మీ దగ్గరికి వస్తున్నారు. సమ్ పీపుల్ హ్యావ్ పర్ఫార్మ్ వెరీ వెల్. అండ్ నెక్స్ట్ టైం నేను ఇంకా నాట్ ఓన్లీ రివ్యూ ఆర్టికల్స్ బట్ ఎక్స్పెరిమెంటల్ స్టడీస్ కూడా ఎక్స్పెక్ట్ చేస్తున్నాను. ఎక్స్పెరిమెంటల్ స్టడీస్ కూడా కొందరు పెట్టాను. దే హ్యావ్ గుడ్ ఫ్యాకల్టీ విత్ యూ, ఆల్ ఆర్ డాక్టరేట్స్ అండ్ దే హ్యావ్ గుడ్ రీసెర్చ్ ఎక్స్పీరియన్స్. I became from, but research experience ki academic experience చాలా difference. Everyone has a research experience. అందరికీ research experience ఉంది. They came from there only. But మీకున్న ఫ్యాకల్టీ మీరు యూటిలైజ్ చేసుకోండి. ఎందుకంటే బయటకు వెళ్ళిన తర్వాత after MSc, why my origin for the PhD is not my MSc. Industrial. ఎందుకు నేను PhD చేయాల్సి వచ్చిందండి? First thing. I joined as an MSc student. After my MSc, I got 3 opportunities. I chose one. I don't want to mention any company. There, one guy is humiliated me. After all, you are an MSc fresher, you don't know anything. In industry, there is one concept, SOP, Standard Operating Procedures. Only that only you have to follow. You don't have any chances to change any kind of makers. Any mistake cheyadam ledhu. You have to follow the SOP. The regular activities, morning vachama, SOP chadavama, SOP lo unna follow ayam. Ante. There is nothing new to learn about it. అక్కడ మీకు కొత్తగా నేర్చుకోవడానికి ఏం ఉండదు. జస్ట్ పొజిషన్ పెరిగే కొద్ది మీకు డెసిగ్నేషన్స్ పెరుగుతాయి అండ్ యాక్టివిటీస్ పెరుగుతాయి అంతే. బట్ ఆఫ్టర్ దట్ పర్సన్ హ్యూమిలేటెడ్ మీ ఇన్ ఫ్రంట్ ఆఫ్ ఆల్. యాజ్ ఎ DM, he should score me some experience. But he humiliated me in front of all of them. So I got thought that I want to take a PhD admission. So I want to go above him. See, right now if I go for the same company as a PhD, like as a doctorate, they directly give me as a DM in that company. But on my track I came to this economic section. So my point of origin is that. So నేను చెప్పేది ఏంటంటే you utilize the options. మీకు చాలా ఆప్షన్స్ ఉన్నాయి. In the third year you have to be in advance. మీరు థర్డ్ ఇయర్కి వచ్చారు కాబట్టి మీరు ఈ పాటికి ఆప్షన్ చూస్ చేసుకొని ఉండాలి. నెక్స్ట్ ఏంటి అనేది. ఎందుకంటే యాజ్ ఏ BSc you won't get. బయట పే స్కేల్ 13,000 మ్యాక్సిమం యాజ్ ఏ BScకి. BSc పే స్కేల్ 13. ఇదే కటింగ్స్, అకామిడేషన్ ఇవన్నీ చూసుకున్నా కూడా యూ వోంట్ గెట్ నథింగ్ టు హోమ్. మీరు ఇంటికి పట్టుకెళ్ళకి ఏముండదు. జస్ట్ సింగిల్ లైఫ్ కూడా బతకడం ఏముండదు. ఎందుకంటే వచ్చే దాంట్లో 5,000 మిగులుతుంది. మీ సేవింగ్స్ ఎక్కడ ఉంటాయి? మీ హాస్పిటల్ని చూసుకోవాలి. ఎవరు టు టేక్ అప్. మనమేం డోంట్ మెయింటైన్, మనం కంప్లీట్ హెల్దీ కాదు కదా. ఎట్ సమ్ పాయింట్ విల్ గెట్ ఎఫెక్టెడ్. అప్పుడు చూసుకోవాలన్నా సమ్ యూ హ్యావ్ టు గెట్ సమ్ సేవింగ్స్. సో నేను చెప్పేది అంటే నెక్స్ట్ టైం ఇలాంటి జరిగినప్పుడు యూటిలైజ్ చేసుకోండి. ఆపర్చునిటీస్ ఏమున్నాయి? ఎవరైనా వస్తున్నారు, వాళ్ళకి తెలిసిన ఐడియా ఏంటి, మనకు తెలిసిన ఐడియా ఏంటి, మనం ఏమైనా వాళ్ళతో వర్కప్ చేయొచ్చా? మేము ఏదైనా కాన్ఫరెన్స్ వెళ్తే ఫస్ట్ థింగ్ ఎవరెవరు ఏం ప్రజెంట్ చేస్తారు అదే అర్థం చేస్తాం. మాకు ఇచ్చిన ప్రోగ్రామ్ స్కెడ్యూల్ చూస్తాం. దాంట్లో ఎవరెవరు ఏం ప్రజెంట్ చేస్తున్నారు, ఎవరెవరు మనకి ఎంతవరకు సూటబుల్ అవుతున్నారు, మనం ఇంటరాక్ట్ అయితే మన ఐడియా నాలెడ్జ్ షేరింగ్ ఎలా ఉంటుంది. సో ఇవి మనం ఫస్ట్ థింగ్. వచ్చాము, క్లాస్ టైంకి వెళ్ళిపోతున్నాం, బయట తిరుగుతాం, ఇదంతా అన్నెసరీ. జస్ట్ 1 day 2 days ఇట్స్ ఎ ప్రోగ్రామ్. రోజు పెట్టి ప్రెజర్ చేయరు కదా. Some people are listening, some people are not listening. The Brahmachari sir has got serious regarding this issue. Okay. So next time ఇలాంటి కాన్ఫరెన్స్ ఎక్కడైనా జరిగితే మీరు ట్రావెల్ చేయండి. ట్రావెల్ చేసినప్పుడు అక్కడ ఎక్స్పీరియన్స్ ఫీల్ అవ్వండి. షేర్ ద నాలెడ్జ్ అమాంగ్ యూ. సో అప్పుడన్నా మనకి థింకింగ్ న్యూ అంటే మనం కొత్తగా థింక్ చేయడానికి ఐడియా వస్తుంది. ఓకే, థ్యాంక్ యూ వెరీ మచ్.
All of you, after 20 minutes. అందరి absent days.
Very good evening to all of you. I, Sri Lakshmi, HOD of Biotechnology and one of the organizing secretaries of this two-day international conference, extend my warm and respectful greetings to all the dignitaries, esteemed guests, faculty members, participants and students present here today. It is my privilege and honor to welcome you all to the valedictory session of our two-day international conference, Future Ready Life Sciences Curriculum, Research and Emerging Technologies, ICFLET 2026, conducted on 23rd and 24th February 2026. Today marks the successful completion of an enriching and insightful event, and we are delighted to celebrate this occasion together. With deep sense of gratitude to our Lord Almighty, who supported us with His immense blessings for the success of the program, I request our brothers, Kramalish and his peer, for a small prayer song.
Oh, give thanks to the Lord, for He is good. Oh, give thanks to the Lord, for He is good. Oh, give thanks to the Lord, for He is good. Yes, eternal is His love. I will sing to my God, never ceasing. All my life I will tell of His wonders. He's the maker of all earth and heaven, and the ocean, the seas, and all they hold. Oh, give thanks to the Lord, for He is good. Oh, give thanks to the Lord, for He is good. Oh, give thanks to the Lord, for He is good. Yes, eternal is His love.
Now, I respectfully invite the dignitaries onto the dais. First, I invite Dr. K. Balachandra, Head Department of Microbiology, convener of this conference, onto the dais. I invite Dr. B. Sivakumar, Vice Principal, First Year UG, onto the dais. I invite Reverend Father Dr. S. Melchior, SJ, Principal of Andhra Loyola College, onto the dais. I invite Dr. Rex Angelo, Correspondent, Andhra Loyola College, onto the dais. I invite our today's guests of honor, Dr. Ashish Rutala, Dr. G. Ashok Chakravarti, and Dr. P. Harikant, onto the dais. It is our immense pleasure to have Mr. S. Tarachand Garu as our chief guest today at Andhra Loyola College for our two-day international conference. Mr. Tarachand Garu is an officer of the Indian Post and Telecommunications, Accounts and Finance Services, IP and TAFS, 1995 batch, Government of India. He is presently serving as Controller of Communication Accounts, Department of Telecommunications, Andhra Pradesh, Vijayawada. Thank you, esteemed dignitaries, for gracing this occasion with your presence. Now, every successful event carries with its moments of learning, interaction, and achievement. I request Dr. B. Sivakumari Garu, Professor in Botany, Vice Principal of First Year UG, to present a report on the two-day international conference conducted in Andhra Loyola College by the Departments of Life Sciences.
Distinguished dignitaries on the dais, respected speakers, the departments, faculty members, research scholars, students, and esteemed participants. Good afternoon to you all. Good afternoon to all you students. Andhra Loyola College stands as a symbol of academic dynamism and progressive vision. It is an institution that is always welcome constructive academic reforms and adopting emerging global trends in education and research. In today's rapidly advancing world, life sciences are undergoing transformational changes driven by emerging technologies, interdisciplinary research, artificial intelligence, biotechnology, sustainable agriculture, and translational medicine. The traditional boundaries between disciplines are dissolving, and there is an urgent need to align curriculum with cutting-edge research and industry expectations. It is with this vision that we felt that there is a necessity to organize an international conference to create a dynamic platform where academicians, researchers, industrial experts, and young scholars could come together and exchange ideas, discuss their new innovations, and collectively shape a future-ready life science ecosystems. Conferences like this are essential to bridge the gap between classroom reading and global scientific achievements. Respected members, it gives me an immense pleasure to present the report of today's International Conference on Future-Ready Life Sciences, Curriculum, Research, Emerging Technologies, organized by the Department of Life Sciences, including botany, zoology, microbiology, biotechnology, food technology, and agriculture, rural development of Andhra Loyola College on 23rd and 24th of February at Bishop Mumadi Hall. On 23rd morning, the conference commenced with a graceful inaugural ceremony. The program began with the blessings by Reverend Father Joseph Bastien, ST, Rector, followed by greetings from Reverend Father Dr. A. Alexangelo, ST, Correspondent, Reverend Father Dr. S. Melchior, ST, Principal, delivered an inspiring message emphasizing academic excellence and research integration in life sciences. The theme of the conference was thoughtfully introduced by Dr. K. Balachandra, convener of the conference, who highlighted the importance of aligning life sciences education with rapidly evolving research landscapes and emerging technologies. A significant highlight of the inaugural session was the release of compendium for abstracts, symbolizing the scholarly contribution and academic richness of the conference. Total, we received 105 registrations from outside the college, and our students, nearly 300 students, are registered for this conference. Total resource persons, we invited 8 persons, total poster presentations 31, and total oral presentations 25 in these 2 days. The inauguration of this conference followed by keynote address. The keynote address was delivered by Dr. M. N.D. Kumar, a research professor from the University of Alabama, United States. He explained the importance of life sciences, and he delivered a talk on curating the future, aligning the life science curriculum with emerging tech and research. Provided profound insights into curriculum modernization, interdisciplinary integration, and the incorporation of new technologies in life science education and research. Afterwards, lead lectures followed by Dr. R.S. Prakasham from CSIR, Indian Institute of Chemical Technology, delivered a talk on mutanase enzyme platforms for next generation dental care and biocontrol formulations, followed by Dr. P. Veerbrahmachari from Krishna University presented a talk on microbial signatures of diet, understanding host microbiome interplay. These lectures stimulated the vibrant academic discussions and broadened interdisciplinary perspectives. The day concluded with parallel sessions of both oral and poster presentations for faculty and the students for Junior Scientist Awards. These sessions showcased the active and also high quality research contributions from the faculty members, scholars and students. On second day, that is on 24th February 2026, the intellectually enriching session started with a webinar. Lead lecture was delivered by Dr. P. Praveen from Miguel University on building cells for cure, engineering stem cells into the therapeutic technologies, followed by Dr. Harikant Paroka from the Fruit Research Station Sangareddy delivered a talk on nutraceuticals in horticultural crops. The conference also featured notable technical presentations. For example, Dr. Nadeem Siddiqui from Yale University presented a paper on Titicon-based 3D porous co-fold for osteochondral tissue remodeling mechanism. Later, Dr. G. Ashok Chakravarti from Malareddy University delivered a lecture on sustainable agriculture for the 21st century, challenges, innovations, and future pathways. Later, Dr. Ashish Gundala from SR University delivered a talk on decoding the metabolic crossroad, a network biology analysis of MET-S and type 2 diabetes. Research presentations on session 3. In the afternoon session, oral presentations for Young Scientist Award and poster presentation for Junior Scientist Award competitions conducted for participants from various colleges and universities. Finally, what is the outcome of conference means? We encouraged the interdisciplinary scientific dialogue between the different disciplines, highlighted the emerging research and transformative technologies, fostered collaboration among the researchers and academicians, provided valuable exposure and recognition to young scholars. Conclusion. The conference was a resounding success, reflecting the collective dedication of the organizing committee, faculty members, participants, research scholars, and student volunteers. On behalf of the Departments of Life Sciences, we express our sincere and heartfelt gratitude to the management of AFC and all the speakers, delegates, sponsors, and contributors for their invaluable participation and support to making this international conference a grand academic success. Thank you once again. Thank you for giving me this opportunity.
Thank you, ma'am, for the comprehensive overview of the program. Now, I request Reverend Father S. Melchior S.J., Principal of Andhra Loyola College, to share his thoughts, observations, and impressions about the conference.
Mr. Tarachand Garu, today's chief guest for the two-day International Seminar Valedictory Program. The guests of honor seated on the dais, Reverend Father Dr. Rex Sanchuru, our beloved correspondent, Dr. K. Balchandra, the convener of this seminar, the HODs of 6 departments, botany, zoology, biotech, microbiology, food technology, and agriculture and rural development. And dear participants from various colleges, various institutions, and dear students, good evening to all of you. How are these 2 days? All of you maintain perfect silence these 2 days? Yeah?
Is it a learning experience for all of you? How many of you feel that these 2 days is a learning experience? How many of you feel that at least you learned something? Very good. Don't look at the other and raise your hand. You have to raise your hand, not the others. Okay? It's a good number of you. I'm happy to know that good number of you have learned something, if not fully, at least something. Even if you have learned a new word, a new vocabulary in this life sciences, purpose is achieved, I feel, because at least you have learned something. So I congratulate the departments. In fact, when I called Balchandra sir and almost mandated him to conduct this seminar, he was perplexed. He is lost. Got frightened to organize. Father trying to find an excuse. I said, no, you are going to do that. And today we see the baby of it. Okay? So congratulations, Balchandra, though you had few initial fears. But with the backup of our beloved Vice Principal Shivakumari ma'am, who is very experienced in organizing these events and who has contacts with many scientists around the country, and other experienced person, Dr. Rosemary ma'am, and committed HODs of other departments and the members of the departments. Today, we can say that these life sciences departments have conducted this 2-day international seminar and a grand success. So congratulations to all of you. And for your cooperation, students, though we call the 6 departments, but the number is small when we put together all of you, but you have proved yourself that even the small number can do well. It's not the number, but it's the commitment and the quality that matters for any event to be a grand success, and you have proved that you are a qualitative departments. So congratulations to all of you. And I also congratulate all the research scholars who have presented their papers here, and I also thank on behalf of the management and the college all the resource persons for these 2 days who have spared their time and came over here and shared their knowledge with their presentations, which enriched the knowledge of our students. Thank you, sirs, for coming over here, and those of you who are not here also convey my wishes to them and thanks to them, to those who have presented here. And I'm more joyful, my heart is overwhelmed when I know some of my students have presented their papers. So congratulations, students, and keep up that research bent of mind, and surely that you would be one day called the scientists. And today, both Father Correspondent and myself were taken up by surprise by 2 of your life science students, who are this guy, come here, and where is Anmol girl? Sujit and Soumya, or no, not you, life science. Sudeep, they are not life science students, no? Yeah, where are they? Yeah, you say one more years, yeah. See, they have brought up a book, yeah, with ISBN number. That's what all of you should cultivate, that habit of it. No, that is what I encourage all of you students. Take interest. It all matters what type of interest and how you mold yourself and how you orient yourself. So congratulations to all of you, and may God bless all of you, and keep up this spirit of research bent of mind. Surely it would yield a positive result to all of you. Keep up that, and my sincere congratulations once again to the 6 departments for putting efforts, having a lot of meetings, clarifying, in the last 3-4 days I made you run up and down, to contact this person, that person, and I know the number of times you went to the CCE because we have organized this two-day international seminar in collaboration with the State RUSA Project Director. So we must thank also the RUSA Project State Director, and also the man behind us is Mr. Sriranga, who helped us, who guided us. So we must give him a big round of applause in this aspect. Who guided us through for this, who made it to become a reality. So thank you. May God bless all of you. Keep it up, this spirit of research. Thank you.
Thank you, Father. Now I invite our chief guest, Mr. S. Tarachand Garu, to motivate our students with his words.
Good evening, all the dignitaries on the stage and all the students present at this today's workshop. I appreciate the efforts of the organizing committee in conducting this workshop on life sciences, future ready life sciences. I would just like to give you a brief about myself. I am purely non-science guy. I am science and so I can speak in Telugu also for few words. Yeah. So I am purely arts, the humanities. I was good in social studies. So science and bhayam. So now you are talking about pure sciences, life sciences. So it is beyond my understanding, but I appreciate a lot of things are happening, a lot of innovations are happening. I thank Mr. Harikant. I know him since last few years from Hyderabad. So he told about this workshop and I'm happy to be here. So I'm a civil servant, UPSC IAS exam. In that I got into Indian Postal and Telecom Accounts Finance Service. So I am from 1995 batch. So I just missed by a few marks. I didn't get IAS or IPS. Discussed, and last 2 days you have all covered many topics about sustainable development, AI, then machine learning, then biotechnology, then digital autonomy, remote sensing technology, and other things. So I am really happy that all the students here have participated actively, and all the research scholars, all the professors and speakers who have come. You had a very good sessions. All the students would have really, got to know many things. And I wish you all the best for your future. You would have planned, what is your plan? I don't know, but as a civil servant, I will tell you, whatever you have planned, please do with your utmost care. Take care of your, whatever dreams you want to achieve. Please strive hard for that, do hard work. There is no easy way. So please do your best. Go for good mentors. Mentorship is very much required. Even when I was preparing for my civil service exam, I required mentorship. That is basically coaching. You require some good tutor or a good coach. So for all of you who are from life sciences, the subject is very vast. So life sciences can play a major, it's playing a major role in the entire, the world or the environment. But at what cost we are doing these things? So the subject you have chosen is like life sciences education for sustainable development. So we have been seeing everywhere development is happening. At what cost? Are we doing at the cost of the environment? Are we doing at the cost of the ecosystem? So we as a researchers, we as a students, we have a responsibility towards the world, towards our environment, to take care of our environment. As a students, as a researchers, and as all the faculty and the resource persons, I hope this 2 days workshop has been a very fruitful discussion, and I couldn't be part of the workshop, but I will definitely take some feedback from the resource persons, and I would like to go through. And my office is in Vijayawada. It is in MG Road. And sir, I am really thankful for calling me as a chief guest for this prestigious institution, which is 72 years old, which was founded in 1953. And recently, I think 70 year celebrations was held. I was aware of that. And I have many friends who have graduated from this college. They did PG from Hyderabad Central University. Many of my friends are there. Maybe I will connect to them. I will send my photos to them. And I say that I am also part of Loyola Andhra College. And I feel it's a very beautiful campus, sir. Really, just entering in this campus, I was really wondering. Such a big campus in Vijayawada, very beautifully maintained. I thank all the organizers for conducting such a very beautiful, for maintaining such a big campus with a lot of greenery, and for conducting this workshop. I thank you for calling me, sir. And it's a learning process for me. So I am like you as a student. I am also learning now. So I wish all of you all the best for your future. Thank you very much.
Thank you, sir. Thanks for the inspiring speech. Now, achievements deserve recognition and excellence deserves celebration, isn't it? So now it's the time for prize distribution. I request Dr. Chand Basha, Assistant Professor in Zoology Department, to announce the prizes.
Before I read out the awards, I have to appreciate the meticulous, Herculean and strenuous task made by the jury, led by Dr. Prakashan sir and Dr. Ashish Mutala sir. Though they have scrutinized number of papers, they kept the fresh event, and they kept the event fresh. And for the students who won the awards, it's just the beginning of something, and who don't, it's their experience. Regarding Junior Scientist Award, that means for poster presentations, there was a tie for the second prize. So we decided to award the second prize for two papers. The first, that means the second prize awarded to first paper, R. Tejaswini and D. Priyadarshini from Siddhartha Mahila Kalasada. The title of the paper is Anti-Narcotic Resistance. Request our chief guest to award the Junior Scientist Award second prize, set one. That means there are two prizes for the second prize. Second prize, another second prize. Paper led by Dinesh Tapikar and Kanti Kumar from Department of Zoology, ANC. And the first prize goes to the paper led by R. Sohail, V. Hemant and C.V. Cherukuri from Geetham University. The title of the paper is Systematic Investigation of Encapsulation Efficiency, Release Behavior and Functional Bioactivity of Pigment-Rich Phytoconstituents. Congratulations.
Bro.
Prashanti from Yogi Vemana University, Kadapa. Title of the paper is Sustainable Phytomediated Synthesis of Iron Nanoparticles from Deodorant. The second prize goes to Sheikh Noor from KU College of Pharmacy, and the title of the paper is Antimicrobial Resistance Patterns of E. coli in India, a systematic review of emerging trends and public health implications.
తర్వాత ఈవెంట్ కనెక్ట్ చేసిన వాళ్ళే పంపిస్తారు. అరే అరే అనమల రాకుండా స్క్రీను.
And finally, the first prize goes to Yadav Srikan from KU College of Pharmacy. The first prize is Anti-Bacterial, and his paper is Indole-3 Carbinol protected the brain from focal cerebral ischemic injury. Congratulations. Thankful to Chief Guest and other dignitary for awarding the awards.
Thank you, sir. Congratulations to all the winners. Your dedication and hard work are truly commendable. Let us give them a big round of applause. Time for felicitations. Now I request our Reverend Fathers, Melchior, Principal of Andhra Layla College, and Dr. Rex Angelo, Correspondent, to felicitate the Chief Guest, Mr. S. Tara Chandra. Meanwhile, I request all the HODs of the departments, members of the departments, and prize winners to assemble near the podium for a photograph after the felicitation.
సార్ షణ్ముఖ్ షణ్ముఖ్ నువ్వు ఆయన పక్కన సార్ ఆయన కూడా తప్పమందా ఆడవాడు పిల్లలు
Participants, sorry, prize winners, please come along with your certificate. Students, please maintain silence.
సైడ్ ఇస్తారా కొంచెం అలాక అదే లోకి లోకి కూర్చోమని చెప్పా అదే ఇంకా వచ్చేసారు కావాలంటే ఆపేసి ఇంటికి వెళ్తాం రావడం ఎందుకంటే తీసుకొచ్చి పెట్టింది అక్కడ అయ్యా నాకు కాల్దా తీసుకొచ్చేస్తుంది నాకు
కెమెరా సర్దేస్తా ప్రశాంతంగా చూస్తుంటా లైవ్లో కట్ చేసి పెట్టి కావాలంటా
Students, please maintain discipline. I invite Dr. K. Balachandra Garu, convener of the program, HOD of Microbiology, to deliver half of thanks to the audience.
Good afternoon to one and all. Respected dignitaries on the dais. Respected dignitaries on the dais, distinguished speakers, esteemed delegates, faculty members, research scholars, students, and dear participants. As we come to the conclusion of the intellectual enriching two-day international conference on future-ready life sciences, curriculum, research, and emerging technologies, it is my proud privilege to propose the vote of thanks. First and foremost, we express our profound gratitude to the management of Andhra Laila College for their constant encouragement, vision, and support in making this international academic event a grand success. We are deeply thankful to our rector, Reverend Father Joe Sebastian S.J., for his blessings and moral guidance. To our esteemed correspondent, Reverend Dr. Father Rex Angelo S.J., for his inspirational words and unwavering support. And to our respected principal, Reverend Father Dr. S. Melchior S.J., for his leadership, motivation, and commitment towards academic excellence. We place on record of our profound appreciation to keynote speaker Dr. M.N.V. Ravikumar, Research Professor and Assistant Vice President for Interdisciplinary Research, Founding Director of the Center for Convergent Biosciences and Medicine, Intern Chair, Department of Translational Sciences and Medicine, the University of Alabama, United States, for his thought-provoking keynote address on aligned life sciences curricula with emerging technologies and research. My sincere thanks to all lead speakers and technical speakers who enriched the conference with their expertise and scholarly contributions, beginning with Dr. R.S. Prakasham, retired Chief Scientist, CSIR Emirates Scientistship, Medicinal Chemistry and Biotechnology Session, Indian Institute of Chemical Technology, Hyderabad, India. Dr. P. Veerabrahmachari, Associate Professor, Department of Biosciences and Biotechnology, Krishna University, India. Dr. Praveen, Research Associate, Stem Cells Bioprocessing Lab, Department of Chemical Engineering, MC Grill University, Canada. Dr. Harikant Porika, Scientist, Horticulture, Fruit Research Station, Sangareddy District, India. Dr. Nadeem Siddiq, Associate Professor, Koneru Lakshmiya Education Foundation, Deemed University, Vodeshwaram, Guntur, Andhra Pradesh. Dr. Ashok Chakravarti, Associate Professor, Department of Agronomy, School of Agricultural Sciences, Malareddy University, Hyderabad, India. Dr. Ashish Runatala, Associate Professor and Assistant Dean, International Relations, Department of Basic Sciences, School of Sciences and Humanities, SR University, Warangal, Telangana. I am thankful to co-conveners and faculty members organizing departments of life sciences, botany, zoology, microbiology, biotechnology, food technology, agriculture and rural development for their meticulous planning, coordination and tireless efforts in organizing this conference. Our sincere thanks to session chairs, reviewers, judges and advisory committee members for their scholarly contributions and fair evaluation for the presentations. We also acknowledge with gratitude the enthusiastic participation of faculty members, research scholars, students, and delegates from various institutions whose active engagement through oral and poster presentations enriched the academic discourse. A special word of thanks to organizing committees, volunteers, technical teams, and non-teaching staff for their behind-the-scenes dedication, efficiency, and teamwork, without which this event would not have been possible. We are also thankful to all who extended support directly or indirectly, contribution to smooth conduct of the conference. Finally, we thank each and every participant for being a part of this academic journey and for making the International Conference 2026 a memorable, successful event. Thank you. I request all of you to stand up for national anthem.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे